At first glance, the water seems empty. Morning light filters through the tangled mangroves of a northern Australian estuary, painting long, shifting bars of gold across the surface. The tide slides in with deceptive calm, and beneath it the water is clear enough to see the silty bottom, the scattered blades of seagrass, the darting shapes of juvenile fish. But something moves here that almost doesn't exist to the human eye. A glimmer, a suggestion of motion that the brain hesitates to identify, because it is not supposed to be there. Then, if the sunlight hits it just right, a faint cube-shaped bell materializes in the water column, refracting light in tiny rainbows. Inside it, slow pulses contract and release like a heartbeat. The box jellyfish is hunting. The species Chironex fleckeri is among the most venomous animals on Earth. It doesn't roar or chase or bare its fangs. Its hunting weapon is chemistry, not claws. Yet in the silent underwater theater of the mangroves, its method is as ruthless and efficient as any apex predator's ambush. The jellyfish drifts on the current, its bell slightly tilted forward, trailing 60 to 80 tentacles that stretch for several meters behind it. Each tentacle bristles with millions of microscopic cells called nematocysts, spring-loaded capsules coiled with harpoons of glass-like protein. When these tiny harpoons sense a chemical cue, touch, or even subtle pressure change from nearby flesh, they explode outward faster than a gun's bullet, embedding into the target's skin and delivering a measured dose of venom that can paralyze small fish almost instantly. To watch this creature hunt is to see chemical precision at its peak. A small anchovy swims too close, unaware of the danger. The jellyfish doesn't lunge, it doesn't need to. The tentacle brushes the fish's flank, and within a few millionths of a second, the harpoons have fired. Ion channels inside the fish's nerve cells are blocked. Muscle control vanishes. The prey freezes mid-swim, shudders once, and begins to sink. The jellyfish curls its tentacle inward in a slow, deliberate reel, bringing the paralyzed fish toward the bell's underside, where it will be digested in the mouth-like cavity. There is no struggle, no chase. Only silence and the unhurried choreography of chemical mastery. Unlike most jellyfish, the box jellyfish is not a passive drifter. Its body architecture grants it something extraordinary in the gelatinous world: control. Each corner of its box-shaped bell holds a complex sensory organ called a rhopalium. Within each rhopalium are clusters of eyes, 24 in total, that include camera-type eyes with corneas, lenses, and retinas capable of forming true images. It can see the world, not just light and dark. These eyes allow the box jellyfish to navigate the maze of mangrove roots, avoid obstacles, and actively hunt schools of fish. In experiments, individuals have been shown to patrol back and forth like sentries, hovering at the edges of light zones where prey congregates. When the tide changes, they retreat deeper into the labyrinth, always keeping themselves aligned with the subtle flow of water that carries food their way. Their transparency is not just camouflage, it is physics perfected for invisibility. The gelatinous tissue refracts light at nearly the same index as seawater, making the animal's body effectively vanish except for the faintest edges. Only when sunlight strikes at certain angles does the bell betray itself as a prism-like shimmer. This optical stealth is an evolutionary adaptation as advanced as the camouflage of any cuttlefish or octopus. It allows the jellyfish to drift in plain sight, an assassin hidden in the glare of daylight. The venom itself is a biochemical marvel. When a human is stung, the effect can be catastrophic. Within minutes, the neurotoxins and cardiotoxins begin attacking the nervous system and heart muscle, opening pores in cell membranes and causing potassium and calcium ions to leak uncontrollably. The result is a storm of pain, muscle spasms, and in severe cases, cardiac arrest. The most dangerous aspect is speed. Victims have been known to collapse before even reaching shore. Yet the venom's design isn't meant for humans. It evolved for fish, whose small size and fast metabolisms demand a venom that kills before escape. In evolutionary terms, the venom is both sword and shield, fast enough to secure prey, strong enough to deter predators. Few creatures are foolish enough to feed on box jellyfish, though sea turtles, protected by thick skin, are one of the rare exceptions. In this mangrove theater, every weapon is chemical. Coral polyps use toxins to fight for space. Sponges leak compounds that deter competitors. Even seagrasses release allelochemicals to defend their turf. The box jellyfish simply turned this molecular warfare into a hunting strategy. Its venom is composed of multiple proteins, each with specialized targets: hemolysins that rupture blood cells, pore-forming cytotoxins that damage tissues, and peptides that hijack ion channels in nerves. Scientists studying these components have found potential in them, not for harm, but for healing. Certain peptides in box jellyfish venom have inspired research into painkillers, as they interact with the same molecular gates that transmit pain signals to the brain. The paradox is striking. A creature that inflicts unbearable pain may one day help silence it. The nematocysts themselves are among the most extraordinary biological machines in existence. Each capsule stores enormous internal pressure, roughly comparable to what's found inside an industrial steam boiler. The trigger, a cilium called a conidocyl, waits for the right stimulus, often a combination of chemical cues specific to prey skin mucus. Once activated, the coiled thread shoots outward at speeds exceeding 30 meters per second, delivering venom through the hollow filament. The entire process unfolds faster than the human eye can register. Under the microscope, it looks like a microscopic explosion, a flash of biological weaponry evolved long before vertebrate predators ever swam these waters. What makes the box jellyfish's precision so chilling is its behavior. Despite lacking a brain, it demonstrates coordinated movement and decision-like responses. The nerve net distributed across its bell synchronizes contractions that allow it to pulse rhythmically, steering with a purpose that seems almost intelligent. It can accelerate when chasing fish and decelerate when retreating to shaded areas. It orients itself toward brightness but avoids excessive glare that could signal shallow water or danger. In laboratory tanks, box jellyfish have been observed learning simple spatial tasks, navigating around obstacles more efficiently over time. Their body may be gelatin, but their control systems rival that of primitive robots engineered by humans. The mangrove shallows, where they thrive, form a chemical and sensory labyrinth. The water is warm, the salinity fluctuates with tides and rainfall, and visibility changes hour by hour. Few environments test survival systems so harshly. Yet the box jellyfish reigns here precisely because of its adaptability. Juveniles begin their lives as polyps clinging to submerged surfaces, roots, stones, or even human debris. When environmental cues shift, usually triggered by rising temperatures and day length, these polyps metamorphose into free-swimming medusae. The timing coincides with the influx of small fish and shrimp larvae into mangrove nurseries, a perfect synchrony of predator and prey cycles. Nature, in this case, has engineered an assassin that emerges when the pantry is full. Field researchers handling these animals must operate with near religious caution. The tentacles, even when detached, retain their firing power for hours. Contact with just a few centimeters can trigger excruciating pain. Vinegar, an acid, is used to neutralize undischarged nematocysts by denaturing their firing proteins, but it cannot reverse venom already injected. Rapid application is critical. Delays of seconds can mean the difference between recovery and collapse. Yet within that danger lies a kind of respect. Understanding the jellyfish's biochemistry is not just about fear, but about decoding one of nature's most refined survival systems. As the tide retreats, sunlight fractures on the rippled surface, and the box jellyfish follows the flow outward toward the open shallows. Its bell contracts and expands with effortless rhythm, maintaining position within a few centimeters of the surface. Fish schools shimmer nearby, cautious but unaware. The jellyfish's tentacles undulate gently, forming an invisible web. One juvenile mullet brushes a strand and goes rigid. The jellyfish reels it in. This is a daily ritual, performed millions of times across tropical coasts from Australia to Southeast Asia. Every pulse, every sting, every kill is part of an evolutionary script written in molecules and membranes rather than bones and teeth. It is easy to imagine the ocean's deadliest predators as sharks or orcas, creatures of muscle and motion. Yet here, in this quiet estuary, death comes from transparency and patience. The box jellyfish doesn't need to move fast. It only needs to exist where everything else must swim. Its venom has evolved for efficiency, so powerful that in evolutionary terms, it borders on overkill. But evolution doesn't design for mercy. It designs for success. For the jellyfish, success means leaving behind offspring that can drift unseen, continuing the invisible reign of a creature that hunts with chemistry, not speed. At night, when the moonlight filters through mangrove branches, their bells glow faintly from refracted starlight. If you were to wade here without protection, you'd see nothing. No fin, no shadow, only the soft tug of tide around your legs. And in that invisible current, perhaps inches away, waits an animal that has mastered the art of pain and paralysis. The water that seems tranquil is, in truth, charged with potential, an unseen network of biological tripwires armed with molecules that can still the strongest heart. In the end, the box jellyfish is less a predator than a living demonstration of nature's chemical intelligence. It has no skeleton, no centralized brain, and yet it moves, hunts and kills with precision rivaling any vertebrate hunter. Its power lies not in strength but in timing, not in aggression but in calibration. To understand it is to confront the fact that survival in the sea is not a contest of size or speed. It is a chemistry experiment that never ends. And in this experiment, the box jellyfish remains one of the ocean's most successful formulas. The sea is calm in the early light, its surface almost metallic beneath the rising sun. Far offshore, where coral shallows fade into open blue, something breaks the water. A smooth, glistening ribbon twisting upward in a single graceful motion. It takes one quick breath, then disappears again without a ripple. The sea snake, the only reptile truly at home in salt water, lives between two worlds, one lung in the air above, the other half of its being coiled around a chemistry of death below. Beneath the surface, its body moves with effortless economy. The flattened tail, evolved into a natural paddle, flicks from side to side, propelling the snake through coral alleys and over sand flats in slow, deliberate arcs. There is no need for haste. Every movement is calculated to conserve oxygen, every curve designed to reduce drag. Though it is a reptile, cold-blooded, air-breathing, scaled, it has become something entirely different here, a hybrid organism shaped by the twin pressures of marine life and toxic mastery. Unlike its terrestrial cousins, it has traded fangs and ambush for stealth and precision, a predator that kills with chemistry before the struggle even begins. From above, the pattern of the banded sea crate or the olive sea snake melts into its environment. Stripes dissolve among the dancing shadows of wave refraction, an illusion perfected over millions of years. Fish and eels pass nearby without alarm, and that is the point. The snake hunts by infiltration. It glides into reef crevices and coral heads where other predators cannot reach, slipping into labyrinths of branching corals where small fish, blennies, and gobies seek refuge. Its head, flat and wedge-shaped, probes into holes, tongue flicking to read faint chemical signatures in the water. What it seeks is not movement but molecules, the invisible scent of life, the chemical whisper of proteins and mucus that betray a fish's hiding place. When the strike comes, it is instantaneous. The snake lunges, delivers a tiny bite, and withdraws almost in the same motion. The fangs, short but needle-sharp, inject one of nature's most sophisticated venoms, a neurotoxic cocktail that shuts down the interface between nerve and muscle. Alpha neurotoxins bind tightly to nicotinic acetylcholine receptors, the molecular switches that tell muscles to move. Once blocked, the message from the brain goes silent. The prey stiffens, then goes limp. There is no struggle, no thrashing. Death arrives with eerie calm, a physiological shutdown that happens before pain can even register. This is biochemical efficiency at its evolutionary peak. Minimal energy expended, minimal risk to the hunter, maximum effect on the prey. For all their reputation, sea snakes rarely bite humans. Their fangs are small, and their venom, though potent enough to kill a grown person, was never meant for us. When they do strike divers or fishermen, it is usually defensive, and often the bite is dry, a warning without venom. The reason is purely evolutionary logic. Venom is costly to make. It requires protein synthesis, metabolic investment, and specialized glands that demand energy to replenish. To waste it on an oversized threat that cannot be eaten serves no purpose. The animal's intelligence lies in restraint. But beneath this measured behavior lies a chemical arms race waged at the molecular scale. The key to sea snake success is their venom's adaptability. Marine prey, with faster metabolisms and smaller bodies than terrestrial rodents, require neurotoxins that act within seconds rather than minutes. Through evolutionary refinement, the venom components, particularly phospholipase A2 enzymes, have been tuned to penetrate fish tissue and spread quickly through capillaries. Some species have developed unique toxins that specifically target marine muscle proteins, making their chemistry a product of both ecological niche and evolutionary specialization. Their venom does not just paralyze, it dismantles. Phospholipases rip apart cell membranes, allowing neurotoxins to reach their targets faster. Metalloproteinases then break down connective tissue, ensuring diffusion. It is a sequence of microscopic assaults, all completed long before the victim's body hits the sand. The physiological adaptations that allow sea snakes to live entirely at sea are as extraordinary as their venom. They have a single, elongated lung that runs nearly the entire length of the body, functioning as both oxygen reservoir and buoyancy regulator. Their skin is semi-permeable, allowing them to absorb oxygen directly from seawater, a form of cutaneous respiration that can provide nearly 1/5 of their oxygen requirements. Salt glands beneath the tongue expel excess sodium with every flick, maintaining balance in a world that would otherwise dehydrate their cells. Even their scales, once rough and protective, have flattened and smoothed, forming a hydrodynamic sheath that reduces turbulence as they swim. The result is an organism engineered not just for survival, but for dominance in a realm once thought inhospitable to reptiles. There is a peculiar beauty to their lethality. The same neurotoxins that arrest a fish's muscles mirror the molecules that control our own. In laboratories, these compounds are studied for what they reveal about the nervous system, how signals pass, how receptors open and close, how synapses maintain precision under electrical chaos. The architecture of antivenom mirrors the architecture of the snake's target. Antibodies designed to intercept toxins before they can lock onto human receptors. Each vial is a synthetic reflection of the sea snake's own chemistry, a counter-design born from the predator's blueprint. What evolution perfected through natural selection, medicine now studies through molecular mimicry. In the wild, sea snakes are more than hunters, they are indicators of the reef's health. They depend on dense fish populations and complex coral structures to thrive. When reefs bleach or collapse, sea snake numbers plummet. Their disappearance often precedes larger ecological losses. In this way, they serve as chemical messengers of environmental distress, sentinels whose absence signals that the ocean's chemistry itself has shifted. Their sensitivity to pollution and temperature makes them both victims and witnesses of climate change. Scientists tracking their populations have noted changes in venom potency across different regions, suggesting that even the biochemical weapons of the sea are evolving in response to warming waters and altered prey availability. One of the strangest aspects of sea snake biology lies in reproduction. Many species are entirely viviparous, giving birth to live young in open water. The embryos develop inside the mother's body, nourished by a placenta-like connection, an evolutionary echo of the mammalian solution. The young emerge fully equipped, miniature replicas already capable of swimming and hunting. Others, like the sea kraits, return to land to lay eggs, a behavior that ties them to shorelines and exposes them to human threats. This dual existence, half land, half sea, illustrates the evolutionary bridge they occupy, a lineage forever walking the boundary between two domains. Even their navigation is chemical. Unlike fish that rely on lateral lines to detect motion, sea snakes sense faint electric fields generated by muscle contractions in nearby prey. The entire body, conducting through seawater, acts as an antenna tuned to biological frequencies. In a coral labyrinth filled with crevices and noise, this sensitivity allows them to home in on the flicker of a heartbeat or the pulse of gills long before visual contact. To the snake, the reef is alive with electrical signatures, a three-dimensional map of moving life painted in currents and ions. When a sea snake rises to breathe, it does so with elegance that borders on ritual. It ascends in a spiral, exhaling the last of its stale air just before reaching the surface, then taking in a deep, efficient breath that fills its entire lung in seconds. It does not linger. Remaining too long risks exposure to seabirds or predatory fish. With the next flick of its paddle tail, it vanishes again, resuming the silent patrol. Between breaths, it can stay submerged for over half an hour, depending on activity and temperature. Its heartbeat slows, metabolism drops, and even its brain tolerates low oxygen levels, a reptilian adaptation refined for a marine stage. On coral slopes, divers sometimes see them weaving among schools of fish, seemingly unbothered by their proximity. It is a sight that defies instinct. One of the most venomous creatures on Earth swimming peacefully among potential victims and indifferent humans alike. Their venom is for precision, not aggression. They kill for need, not impulse. And that distinction, so often blurred in human imagination, is what makes them more fascinating than frightening. The ocean does not waste energy on cruelty. It designs for balance. At the cellular level, the neurotoxins of sea snakes and the synapses of their prey are locked in an evolutionary dance. Each new mutation in a receptor protein, each tiny alteration in the binding site of an ion channel, triggers counter-adaptations in venom composition. It is an arms race conducted at molecular scale, invisible yet relentless. The fish that evolve resistance survive to reproduce. The snakes with slightly altered toxins reclaim their advantage. Over time, both become more refined, more intricate, more dependent on one another's evolution. The predator's weapon and the prey's defense shape each other like sculptor and stone. As the sun sets and shadows stretch over the reef, the sea snake glides out from its coral maze into open water. Its scales catch the last traces of orange light, then vanish into the deepening blue. In this twilight zone, it moves like a ribbon of mercury, silent, weightless, utterly adapted. Its breath belongs to the sky, its chemistry to the sea, its life to the thin border where the two meet. Everything about it, from its salt glands to its synapse-hacking venom, is a testament to evolution's ability to rewrite boundaries. It is not a snake that learned to swim. It is an ocean predator that still remembers the air. Here, the war for survival is fought not with violence but with molecules. The sea snake is both assassin and alchemist, its power drawn from the very reactions that make life possible. Its venom is a lesson in precision, its body a study in adaptation, its existence proof that even cold-blooded creatures can master the chemistry of breath, salt, and death in one seamless design. The ocean, in its vast experimentation, has produced many killers, but few so refined, so balanced, and so quietly lethal as the serpent that hunts with lungs and neurotoxins in perfect harmony. In the shallow waters of tropical reefs and tidal flats, where sunlight fractures into 1,000 liquid mirrors, danger often wears the disguise of stillness. Sand ripples gently with the tide, corals breathe in slow rhythm, and small waves hum across the seagrass beds. It is a world of calm, yet every patch of coral rubble hides an ambush. Somewhere beneath the fine grains of sediment, a shape waits, irregular, motionless, indistinguishable from the surrounding rocks. A crab scuttles past, a small goby darts through, and nothing moves. The ocean itself seems to exhale and hold its breath. Then, from that rock-like lump, a set of eyes flick open, round and unblinking. The stonefish is awake. To the untrained eye, Synancea verrucosa, the reef stonefish, is the perfect ghost. Its camouflage is so exact that even seasoned divers often hover inches away without realizing it. Algae, coral fragments, and silt cling naturally to its skin, which bristles with wart-like projections and crusted folds that resemble eroded limestone. The illusion is not passive, it is chemical. The fish's skin secretes antifouling compounds that prevent the growth of barnacles and parasites, maintaining the perfect rough texture without the giveaways of true decay. This biochemical upkeep ensures that its disguise never rots into uselessness. Evolution, in this case, did not just craft a visual deception, it engineered a self-cleaning one. The stonefish's world is the reef shallows and intertidal flats, a habitat of extremes. 1 hour, it is bathed in water rich with oxygen and life. The next, the tide retreats, leaving it stranded in heated pools where oxygen drops and salinity spikes. Few animals tolerate such swings. The stonefish does so effortlessly, slowing its metabolism, breathing through its skin and gills in equal measure, and sealing moisture within its mucus coat. It can survive out of water for nearly a full day. That endurance is not resilience, it is ambush persistence. The fish will endure anything, even desiccation, as long as it can remain undetected. Its weapon, however, lies not in teeth or speed, but in the 13 dorsal spines that line its back. Each spine is hollow, a natural hypodermic needle connected to a paired venom gland beneath the skin. When pressure is applied, whether by a predator, a careless foot, or the crush of sand, the glands contract and inject venom upward through the spine. It is a system of mechanical precision, a biological syringe that activates only when stepped on. The reaction is defensive, not offensive, but its efficiency has made the stonefish one of the ocean's most feared creatures. The venom is an alchemy of protein toxins designed to inflict immediate and overwhelming pain, to shock a much larger organism into release or retreat before the fish itself is harmed. Inside this venom lies a molecular choreography. Cytolysins, proteins that form pores in cell membranes, create microscopic holes through which ions flood uncontrollably. The result is cell rupture, inflammation, and tissue necrosis. At the same time, other peptides target the nervous system, triggering pain receptors and causing blood pressure to plummet. For small predators such as eels or reef sharks, a single sting can be fatal. For humans, the experience is indescribably excruciating. Victims report the sensation of their limb being torn apart from within, followed by swelling, paralysis, and shock. Yet despite its power, the venom is delicate. Its proteins are heat labile, destroyed by high temperature. That is why hot water immersion, often recommended as first aid, works. The heat denatures the protein structure, neutralizing its molecular function. It is one of the few cases in nature where pain's remedy mirrors its cause, temperature-altering chemistry, just as chemistry inflicted pain. Underwater, the stonefish remains invisible until it is too late. Its hunting style is as brutal as its defense is subtle. Lying motionless, it waits for a fish or shrimp to swim within range. Then, in a motion faster than the blink of an eye, it expands its mouth cavity, creating a vacuum that sucks in the prey whole. This suction strike happens in less than 1/1000th of a second, among the fastest feeding mechanisms recorded in vertebrates. The jaws snap shut, the gill covers flare, and the fish returns to immobility before any ripple betrays the event. There is no chase, no struggle. The reef's most venomous animal kills by stillness, not motion. Evolution sculpted this lethality through the pressures of the reef environment, a place where visibility is high, competition fierce, and energy conservation critical. Speed costs oxygen, stillness costs nothing. The stonefish's heart rate can drop to nearly dormant levels as it lies in wait. Its eyes, elevated slightly above the skull, provide binocular vision while the rest of the body remains buried. The lateral line along its flanks senses faint water vibrations, allowing it to detect movement even in darkness or murky conditions. When a shadow passes overhead, it assesses shape and speed, distinguishing a predator from mere drift. It will not waste venom unnecessarily. The syringe deploys only when the body is pressed. This restraint defines its ecological logic. Venom is not abundance, it is currency. The chemical arsenal of the stonefish has drawn scientific fascination for decades. Within its venom lies stonus toxin, a unique protein complex that ruptures blood cells and causes cardiovascular collapse. Researchers studying this compound discovered that its pore-forming mechanism resembles bacterial toxins, an example of evolutionary convergence where similar chemical strategies evolved independently in vastly different organisms. Nature, in both microbes and fish, found the same molecular solution for defense: make a protein that punches holes. But what makes the stonefish venom truly remarkable is its layered composition. Alongside the lethal cytolysins are analgesic peptides that paradoxically dull pain in the fish's own tissues, preventing it from harming itself when venom glands contract. It is a biochemical firewall, one that allows it to store its own poison without self-destruction. The interplay between the stonefish and humans tells another story: how evolution's adaptations intersect with human fragility. In the Indo-Pacific, coastal fishermen occasionally step on buried stonefish while wading through tide pools. Local traditions once viewed these encounters as curses. Only later did medical science reveal the biochemical truth. Antivenoms developed from immunized horses now save lives, but even so, the pain remains legendary. Divers describe it as a heat that climbs the body, a pulse that synchronizes with the heartbeat until the entire limb throbs like molten metal. It is chemistry translated into sensation, a reminder that in this environment, camouflage does not mean peace. It means potential violence waiting to be triggered. There is irony here. The same venom that terrifies swimmers has potential in medicine. Scientists are studying its peptides for use in treating cardiovascular diseases, as certain components can induce controlled vasodilation, relaxing blood vessels without causing cell death. Another class of molecules shows promise as painkillers, capable of modulating nerve receptors with extraordinary specificity. What was once a weapon of defense may, through research, become a tool of healing. The boundary between poison and cure, as the stonefish illustrates, is one of dosage and context. From an ecological standpoint, the stonefish occupies a subtle but crucial niche. As an ambush predator, it regulates populations of small reef fish and crustaceans, maintaining balance within the ecosystem. Its presence keeps herbivorous species from overgrazing algae, indirectly protecting coral health. Predators that can safely eat it are few: moray eels, large rays, and certain sharks immune to its venom. For them, the stonefish is a high-risk, high-reward meal. The evolutionary standoff between these predators and the stonefish's defenses exemplifies nature's chemical equilibrium. Every toxin invites a countermeasure. Every defense breeds resistance. Even its reproduction carries echoes of chemistry and stealth. During spawning, males and females release eggs and sperm simultaneously into the water column, timed with moonlight and tide. The adhesive eggs sink and attach to sand or coral, where the embryos develop independently. The larvae hatch transparent, nearly invisible, and drift with plankton until they settle. Within weeks, they begin developing the warted skin and cryptic pigmentation that will define their adult camouflage. The metamorphosis from translucence to opacity is gradual, a slow transition from open vulnerability to hardened invisibility. At low tide, when the water recedes and the reef floor bakes under tropical sun, a stranded stonefish might look like nothing more than a lump of coral left behind by the sea. Yet under that inert skin, cells pulse with quiet activity. The mucous barrier traps moisture, enzymes slow metabolism, and the gills pulse just enough to move oxygen from the air. The fish will wait out the heat, sometimes for hours, until the next tide rises to reclaim it. Few animals embody patience so completely. Its entire biology is an argument for stillness as a weapon. As the tide floods back in, the stonefish sinks deeper into the sand, leaving only its eyes visible. Small shrimp hover nearby, oblivious. The water glows gold with sediment stirred by the returning current. Somewhere above, a heron lands, probing the shallows for prey, but not here. The bird senses something wrong with this patch of reef. Even without understanding, instinct warns it away. Below the surface, the rock remains motionless, spines folded, venom glands loaded, chemistry poised. The illusion is complete. The stonefish does not move because it does not have to. Its power lies in the physics of pressure, in the biology of disguise, and in the chemistry of pain. It is a reminder that survival in the ocean is not about motion or intelligence. It is about balance between visibility and invisibility, between lethality and restraint. The world's most venomous fish is also one of its quietest. Its story is not one of aggression, but of patience so profound that even time seems to slow around it. In the hierarchy of marine bioweapons, the stonefish represents perfection through simplicity. No chase, no struggle, no excess energy wasted, only chemistry, pressure, and waiting. The reef around it teems with color and movement, but the deadliest creature here is the one pretending to be nothing at all. Evolution carved its weapon into its spine, wrote its defense into its skin, and concealed its presence beneath a veil of sand. The ocean's most venomous fish does not advertise its power. It hides it, perfectly still, until something careless enough to test the illusion learns the truth the hard way. In the turquoise shallows of the Caribbean, the morning light spills through the water like liquid glass, scattering ribbons of gold across a reef once ruled by balance. Schools of damselfish and wrasses weave through coral gardens, parrotfish grind at algae, and cleaner shrimp attend their silent clientele. But drifting through this harmony comes a vision so ornate it almost seems sculpted, striped fins splayed like silk fans, body gliding in slow, regal motion. It is a creature of extraordinary grace, yet its elegance hides a quiet conquest. The lionfish, Pterois volitans, glides above the coral with the poise of royalty and the appetite of an invader. It does not stalk or chase. The lionfish moves with the deliberation of something that knows it cannot fail. Its fins, elongated into feathery plumes, undulate slowly in the current. Each fin ray tapers into a venom-tipped spine, hollow, pressurized, and ready to inject. The pattern of red, brown, and white bands pulses like warning flags, yet on reefs that evolved without them, there are no creatures left alive that understand the signal. The lionfish's danger is written in color, but the script is foreign to its new dominion. Native to the Indo-Pacific, lionfish evolved within ecosystems that recognized their cues. Predators avoided the distinctive stripes, prey respected the slow looming threat. But when humans unknowingly introduced them to the Atlantic and Caribbean in the late 20th century, most likely from aquarium releases, the balance shattered. Here in these naive waters, their toxins met no countermeasures, their display no deterrent. Within decades, the invader multiplied exponentially, spreading from Florida to Brazil and across the Gulf of Mexico. What began as a handful of ornamental fish became one of the most successful marine invasions in recorded history. To understand the lionfish's rise, one must look closer, beyond the aesthetic. The elegance of those fins conceals weaponry, fine-tuned by evolution. Along the dorsal, pelvic, and anal spines lie venom glands encased in thin sheaths of skin. When threatened or grasped, the sheaths rupture and the venom is drawn up the hollow spines by capillary action, much like oil rising through a wick. The delivery system is purely mechanical, powered not by aggression but by contact. It is a defense that punishes curiosity. A reef shark's probing bite, a moray eel's snap, or a diver's careless touch can all trigger the same reaction, a flash of pain so intense it feels electrical. The venom itself is a complex mixture of thermolabile proteins, molecules sensitive to heat. Once injected, they open ion channels in pain-sensing neurons, flooding them with calcium and sodium ions, triggering an explosion of nerve signals interpreted by the brain as searing agony. Blood vessels dilate, tissue inflames, and the local immune response amplifies the pain cascade. Yet in evolutionary logic, this suffering has purpose. The predator learns, and the lionfish survives. Because these proteins denature at high temperatures, hot water immersion neutralizes them, a simple but effective first aid that transforms biochemistry into manageable biology. Yet the lionfish's venom is only half the story. The rest of its power lies in how it stays immaculate. The same fins that bristle with toxins are coated in a mucus rich with antimicrobial compounds. These secretions discourage colonization by bacteria and parasites, and even foster symbiotic microbes that help keep the skin clean. In a sea where every surface is a battlefield of biofilms and fouling organisms, the lionfish remains spotless. Its scales gleam unmarred, its stripes sharp and pristine. This partnership of chemical and microbial maintenance allows it to thrive in foreign waters, where many species falter under the burden of unfamiliar pathogens. Its hunting strategy is an evolutionary inversion of its defense. Slow, hypnotic, yet devastatingly effective. The lionfish uses its broad pectoral fins like herding sails, spreading them wide to corral small fish and shrimp against rocks or coral heads. Then, with a sudden forward thrust, it opens its mouth in a vacuum-driven strike, sucking in prey faster than the eye can follow. The maneuver is silent and surgical, relying not on pursuit but on deception. Many prey species, confused by the undulating fins and slow approach, fail to recognize the lionfish as a predator until it is too late. In underwater footage, slowed to a fraction of real time, the attack looks like an inhalation of life itself. The prey simply vanishes into the widening gape. Each kill feeds more than just one fish. Lionfish breed continuously throughout the year, with females releasing tens of thousands of eggs in gelatinous, toxin-laced sacs that drift on currents. These floating rafts of embryos deter predation long enough to disperse widely, seeding new colonies across entire coastlines. The reproductive output is staggering, and the juveniles mature quickly, reaching sexual readiness within a year. On reefs already stressed by warming and pollution, such fertility becomes an ecological sledgehammer. Native predators, unadapted to the lionfish's venom and warning patterns, rarely attempt to eat them. As a result, populations boom unchecked, consuming the small reef fish that maintain algae balance. Without these grazers, algae smother coral skeletons, choking out oxygen and light. The chemistry of one fish ripples outward, rewriting the metabolism of an entire reef. The lionfish's biochemistry extends into its invasion ecology in subtler ways. Its mucus, for example, not only protects it from infection, but also appears to carry compounds that deter cleaner fish from approaching. In the Indo-Pacific, native cleaners learn to tolerate this signal, but in the Atlantic, the lionfish effectively exempts itself from the reef's sanitation network. Without cleaners, parasites that might hinder other predators fail to slow it down. Its immunity is both chemical and ecological, an armor woven from molecules and ignorance. Ironically, humans, its only true threat in the Atlantic, are beginning to turn that chemistry against it. Divers now hunt lionfish as both sport and conservation, and the flesh, devoid of venom, is edible and even prized. The spines, once deadly, are trimmed before preparation, and hot water ensures the toxins lose potency. In kitchens and coastal restaurants, the invader is slowly being transformed into cuisine. It is an inversion worthy of evolutionary irony. A species that conquered the Caribbean through venom may one day be controlled by appetite. But beneath that irony lies a sobering truth about adaptation. The lionfish was not engineered to destroy ecosystems. It was built to survive competition in one of the world's most crowded biomes, the Indo-Pacific reef. Its venom, its mucus, its fin display, all were honed over millennia to maintain balance in a community of wary, well-armed fish. It became invasive not because of aggression, but because of absence. Absence of predators, absence of recognition, absence of checks that evolution never needed to provide. Its success is a mirror held up to our disruptions. The lionfish thrives not in harmony, but in the ecological vacuum left behind by overfishing and reef degradation. To study its physiology is to appreciate the precision of its adaptations. The venom apparatus evolved from ordinary fin rays, hard, bony supports modified into hollow conduits. Muscles at the base of each spine compress the venom gland upon impact, injecting measured doses. The same fin pattern that dazzles observers also creates hydrodynamic lift, allowing the lionfish to hover almost motionless without expending energy. It combines aerodynamics, chemistry, and strategy into a single seamless system, a living demonstration that evolution never wastes elegance when efficiency will do. Underwater, in slow motion, the lionfish is hypnotic. Sunlight catches the translucent membranes between its spines, each filament flickering like a banner in slow current. Beneath the surface, beauty, a hidden economy unfolds. Chemical reactions in glands, microbial negotiations in mucus, osmotic exchanges across scales. Even its respiration is precise. Gills optimized for low-flow environments, blood hemoglobin tuned for warm tropical oxygen levels. It is a creature built not for dominance, but for mastery of small advantages, and together, those advantages have made it an ecological empire. Divers encountering lionfish in the Atlantic often describe a strange duality, attraction and alarm. The animal's composure feels almost theatrical, as though aware of its immunity. It doesn't flee when approached, doesn't dart into crevices like other reef fish. Instead, it hovers, turns slightly to face the intruder, and spreads its fins wider, an underwater peacock pose that says both, "Look at me," and "Don't touch." It's a gesture of control, a chemical bluff backed by molecular truth. Every inch of its display is armor made visible. In the larger narrative of marine evolution, the lionfish represents a recurring theme, that beauty and danger are often two faces of the same adaptation. Its biochemistry is not cruelty, it is currency. Venom, mucus, color, and motion are the language through which the species negotiates survival. The tragedy of its invasion is not that it kills, but that it kills so efficiently in a world unprepared to understand it. Its success is the failure of ecological literacy, a mismatch between evolved warning and naive reception. As the light fades over the reef, the lionfish drifts higher in the water column, fins glowing faintly in the dusk. Tiny fish, silhouettes against the dim coral, scatter at its approach. It hovers, calculates, then strikes. Another small life disappears into its maw, another pulse of chemistry sustains its reign. The spines fold, the fins settle, and once more it becomes the motionless ornament of a wounded ecosystem. Grace incarnate, danger distilled. In this chapter of the ocean's arms race, the lionfish stands as proof that the deadliest weapons are often the most beautiful. Its barbs deliver venom, its mucus breeds immunity, its colors deceive, and its calm movement hides conquest. It is both sculpture and system, a living contradiction of elegance and invasion. Evolution gave it beauty to survive. Human ignorance turned that beauty into domination. And as long as it drifts through these waters, silent and unchallenged, the chemistry of one species will continue to reshape the sea. In the shallows of a Japanese bay, the water is glassy and motionless. The tide slides over fine sand and seagrass, carrying whispers of silt that rise and fall like dust in sunlight. Here, among the calm, a small fish drifts, round-bodied, unassuming, moving with a hesitant pulse of fins. Its eyes are large and expressive, its mouth small, its motion almost comical. Nothing about it suggests danger. Yet this tiny bobbing creature, the pufferfish, is one of the most chemically armed organisms on Earth. Its defense is invisible, tasteless, and absolute, a molecule so potent that one drop could silence the body's entire electrical system. The puffer's story begins not with aggression, but with partnership. Deep within its tissues, in the folds of its liver, skin, and reproductive organs, live microscopic allies, bacteria of the Vibrio and Pseudomonas genera. These microbes manufacture a neurotoxin known as tetrodotoxin, one of the deadliest substances known to biology. The fish itself does not synthesize the poison. Instead, it harvests and stores it, turning bacterial metabolism into a living arsenal. Through a biochemical pact born of evolution, the puffer becomes both host and weapon, carrying a symbiotic chemistry that defines its survival. Underwater, the puffer's life is quiet but precise. It spends much of its time grazing on small invertebrates, shell fragments, and algae, using its beak-like teeth to scrape coral or crush snails. Its round shape belies a delicate engineering, an inflation system that can transform the fish into a floating sphere. When threatened, muscles along its abdomen contract rapidly, drawing water into a specialized elastic stomach until the body triples in size. The effect is immediate and unnerving. A predator expecting an easy meal finds a spiny balloon instead, one too large to swallow and possibly lethal to touch. It is an optical and chemical illusion combined. Size deters the strike. Toxin ensures the lesson is remembered. Tetrodotoxin operates at the cellular level, where nerve meets muscle. Every heartbeat, every blink, every breath depends on the flow of sodium ions through voltage-gated channels in nerve membranes. Tetrodotoxin locks those gates shut, preventing signals from traveling. Muscles go quiet, lungs fail, the heart stutters to stillness. Unlike many poisons, it causes no convulsions, no bloodshed, only silence, a paralysis so complete that the victim remains fully conscious while their body forgets how to breathe. A few milligrams are enough to kill a human. Yet inside the puffer, this molecular assassin coexists harmlessly, held in tissues that the fish's own physiology has rendered immune. Its sodium channels, subtly altered by evolution, are resistant to the toxin's grip. Nature, it seems, has rewritten its own locks to carry the key without harm. Not every puffer is equally deadly. Across the vast Indo-Pacific, toxin levels vary with geography and diet. In some regions, puffers are nearly harmless. In others, they are miniature biochemical mines. The reason lies in the bacteria's distribution. Puffers that feed in toxin-rich food webs, consuming crustaceans or worms already harboring tetrodotoxin-producing microbes, accumulate higher doses. In captivity, when raised on sterilized diets, puffers gradually lose their toxicity. Their defense fades not by mutation but by starvation of their microscopic partners. The fish itself is a shell for chemistry it does not fully control, a reminder that in the ocean, individuality often dissolves into shared biology. The partnership is ancient and intricate. Scientists believe it may have begun as an accidental ingestion, fish consuming bacteria-laden prey that provided an unintended layer of protection. Over generations, those individuals carrying bacteria that produced tetrodotoxin gained an advantage. Predators learned to avoid them. The relationship stabilized, and evolution did the rest. The bacteria gained a permanent host, and the fish inherited immunity. Today, their genomes bear traces of this alliance, a coevolution written in molecular tolerance and mutual benefit. In the vast chemical web of the sea, the puffer and its microbes are a single system, not two separate species. For predators, the lesson is immediate and final. Sea snakes that share puffer habitats have evolved sodium channels with small genetic modifications that reduce tetrodotoxin binding, a microscopic arms race played out at the interface of evolution and chemistry. Some species of tiger sharks can also survive mild exposure, their digestive systems breaking down toxins more efficiently than most. Yet few predators risk a second encounter. A single bite delivers enough warning. In tropical shallows, where one mistake can end an evolutionary line, memory becomes as powerful as venom. For humans, the puffer's toxin has become both fascination and peril. In Japan, it is known as fugu, a culinary gamble served in thin, translucent slices. The preparation of fugu requires extraordinary precision. Chefs undergo years of training and licensing to learn how to remove the liver, ovaries, and intestines where the toxin concentrates. Even a trace of contamination can be fatal. Even Yet the demand persists, not in spite of the danger, but because of it. The experience carries an element of ritual, a brush with mortality wrapped in trust for human skill and molecular restraint. In tiny doses, the toxin produces tingling numbness and euphoria, sensations that flirt with paralysis, but stop just short of disaster. It is chemistry as thrill, evolution repurposed into art. From a biological perspective, the puffer fish's relationship with its toxin is a masterclass in balance. Too little, and the fish loses its deterrent. Too much, and it risks poisoning itself or its offspring. During reproduction, females transfer small amounts of tetrodotoxin into their eggs, protecting the embryos from predation without endangering development. This maternal dosing is precise, chemical inheritance measured in molecules. Newly hatched larvae carry enough toxin to discourage small predators, but not enough to interfere with their own physiology. As they grow, their bacterial symbionts re-establish the full defensive concentration, completing the cycle. The puffer's adaptations extend beyond chemistry. Its eyes, positioned high on the head, provide nearly panoramic vision, allowing it to detect threats from any direction. Its skin, elastic yet tough, can withstand the internal pressure of inflation without tearing. Its digestive system filters toxins selectively, concentrating them in safe compartments. Every organ, every process is optimized for coexistence with poison. The fish is not simply immune, it is designed to live symbiotically with danger, embodying the paradox of life that thrives through death. Even the toxin's origin story reaches beyond the puffer. Tetrodotoxin appears across the tree of life, in blue-ringed octopuses, in certain newts, in ribbon worms, and in xanthid crabs. In each case, the chemical structure is the same, a molecular coincidence produced by unrelated bacteria that colonize different hosts. It is an example of convergent evolution at the microbial scale, a universal template for defense reused by evolution wherever it works. The sea, vast and chemically diverse, repeatedly rediscovers the same answer to survival. Stop the nerves, stop the motion, stop the threat. In ecology, puffers play a subtler role than their fame suggests. Many species are grazers that help regulate algae and detritus growth, preventing reefs and seagrass beds from becoming smothered. Their beak-like mouths break down shells and recycle calcium carbonate into sediment, subtly reshaping coastal environments. Yet their presence also enforces an invisible hierarchy. Predators learn avoidance, shaping the behavior of entire fish communities. In places where puffers are abundant, smaller fish forage closer to them, using their chemical protection as an umbrella of safety. The toxin, in effect, becomes an ecological barrier that defines where life can safely exist. The chemistry that protects the puffer has begun to interest medicine. Tetrodotoxin's precision, its ability to block nerve signals without damaging tissue, makes it a promising tool for research into pain management. In minute-controlled doses, it can silence hyperactive nerve pathways responsible for chronic pain and certain cancers. What once killed with indifference may one day heal through understanding. The molecule's cruelty is not inherent, it is simply concentration and context. Evolution builds the tool, humanity decides the purpose. In the clear shallows of the Pacific, a puffer drifts above a coral outcrop. A shadow passes overhead, a heron searching for its meal, but the bird moves on. Beneath the water, the fish continues unbothered, its body glinting under the sun. Around it, countless lives move in quiet choreography, unaware that this small, slow swimmer carries enough chemical power to paralyze an entire reef. Yet the puffer is not a killer. It does not hunt with venom or lash out in anger. Its power lies in potential, in the silent agreement that danger need not be demonstrated to be respected. As the current shifts, the puffer glides toward deeper water. Its skin ripples with light, its eyes scanning, its body a delicate compromise between vulnerability and defense. Inside, bacterial partners go on with their invisible labor, manufacturing molecules that no predator dares to test. The fish is a host, a collaborator, and a survivor, all defined by chemistry that is not its own. The pufferfish reminds us that in the ocean, power is rarely loud. It is silent, microscopic, and shared. Life's greatest weapons are often borrowed, their origins hidden in unseen partnerships. And in that invisible alliance, between bacteria and fish, between molecule and muscle, evolution proves once again that survival is not about domination, but about the quiet genius of coexistence. At first glance, a sea urchin seems harmless, little more than a living pincushion scattered across the ocean floor. In the soft light that filters through shallow reefs, their spines gleam like polished obsidian, arranged in perfect symmetry. They move so little that most divers mistake them for plants or shells, part of the scenery rather than living creatures. Yet beneath this stillness lies one of the most complex defensive architectures in the marine world, a fortress of calcium and chemistry, of spines that sting and microscopic jaws that bite. The sea urchin is a paradox of quiet menace, a slow-moving engineer whose armor hides intricate biological machinery and weapons scaled to both its size and its enemies. The reef around it is restless. Crabs skitter through coral debris, wrasses dart above, and waves shimmer overhead. The urchin remains unmoved, its shadow static against the substrate. Its design is ancient, evolutionary blueprints dating back over 400 million years. While fish evolved fins and sharks refined speed, the urchin perfected stillness and defense. Its entire body is an exoskeleton called a test. a rigid globe of interlocking calcium carbonate plates fused like a puzzle. This shell does not simply enclose. It supports, protects, and when damaged, repairs itself with astonishing chemical precision. Every spine that crowns its body is both armor and instrument. Anchored by ball and socket joints, the spines swivel in all directions, giving the animal a strange, slow mobility as it rolls or creeps across the seabed. At the microscopic level, each spine is built from calcite crystals laced with magnesium, a molecular reinforcement that prevents fracture under pressure. When a spine breaks, as they often do under waves or the curiosity of passing predators, the urchin immediately begins repair. Specialized cells deposit new calcium carbonate in layers, guided by organic proteins that direct crystallization. The new spine grows along the same axis as the old, the magnesium chemistry ensuring rapid hardening and resilience. This process unfolds invisibly, but it is a marvel of self-healing engineering, a stone fortress that mends its own walls without pause. But defense in the urchin is not limited to structure. Hidden among the spines are thousands of pedicellariae, minute pincer-like organs, each no larger than a grain of sand. Under magnification, they resemble tiny robotic arms, tipped with 3 sharp jaws that snap shut in perfect coordination. These are the urchin's microjaws, a mobile minefield capable of reacting faster than its spines can move. Their function is not just mechanical, it is chemical. Many species coat their pedicellariae with venom, a cocktail of neuroactive compounds that inflame tissue and numb pain simultaneously. This duality, burning and dulling at once, is the hallmark of their defense. To a fish that brushes against them, the effect is immediate confusion. The nerves misfire, muscles spasm, and the predator retreats, uncertain of what has struck it. Nowhere is this weaponry more evolved than in the flower urchin, Toxopneustes pileolus, one of the most dangerous echinoderms in existence. Its pedicellariae are larger and more active than most, armed with a potent venom that can cause paralysis or even respiratory failure in humans. The toxins include contractin A, which disrupts calcium flow in muscle cells, and hyaluronidase, which breaks down connective tissue to spread the effect. Yet the flower urchin's appearance betrays nothing. It glows with pastel hues, its spines delicate and petal-like, inviting rather than warning. The irony is sharp. The most beautiful urchin is also the deadliest, a recurring theme in the ocean's evolutionary narrative. In contrast, reef grazers such as the long-spined diadema species are chemical minimalists. Their defense is physical, slender black spines that break on contact, embedding themselves in a predator's mouth or skin. Each spine surface carries mild venom that causes swelling and pain, but its true purpose is mechanical deterrence. The fracture itself becomes the weapon. Because the spines are hollow and brittle, they snap under stress, leaving tiny splinters that continue to irritate for days. For large predators, such encounters are memorable enough to prevent a second mistake. For smaller ones, the lesson is fatal. Between these extremes lies a diversity of strategies. Sand urchins, buried just beneath the surface, rely on camouflage and shallow spines coated in sticky mucus toxins that trap small parasites or deter crustaceans. Rock-boring urchins wield blunt spines for excavation, their pedicellariae tuned more for cleaning than for combat. In every form, defense and maintenance merge. The microjaws constantly groom the urchin's body, removing debris and fouling organisms. This self-grooming is critical. Any growth of algae or encrusting life could interfere with respiration or feeding. The pedicellaria taste surfaces chemically, identifying unwanted hitchhikers by molecular signatures. When detected, they close instantly, gripping and tearing away invaders. The result is an organism that functions like a living coral cleaning crew, its own hygiene system operating around the clock. Beneath this armory, life continues at a measured pace. The urchin's mouth, known as Aristotle's lantern, is a mechanical masterpiece of 5 interlocking teeth driven by muscular plates. It scrapes algae, bores into rock, and grinds coral skeletons into powder. Every bite contributes to the recycling of calcium carbonate in reef ecosystems, transforming solid stone into sand. The beaches of tropical islands, gleaming white and soft underfoot, owe much of their existence to centuries of urchin grazing. What looks like stillness is in fact slow, persistent labor, geological work performed by a creature smaller than a human hand. When viewed through the lens of ecology, the sea urchin is both creator and destroyer. In healthy numbers, it maintains balance, preventing algae from smothering coral. When predators such as sea otters or triggerfish decline, urchin populations explode, leading to urchin barrens, vast stretches of reef stripped of vegetation, reduced to lifeless rock. The chemistry that once protected the urchin becomes ecological tyranny. Yet nature always seeks balance, and predators adapted to the spines, like the wolf eel and the California sheephead, are slowly reclaiming their roles in some regions. The dance between venom, armor, and appetite continues, a living negotiation of energy and survival. From a material science perspective, the urchin's skeleton offers lessons in design. Its calcite structure is not uniform. Under scanning microscopes, the crystals reveal a porous, foam-like pattern that distributes stress evenly. This makes the test lightweight yet strong, a principle now studied by engineers developing impact-resistant ceramics. The incorporation of magnesium alters crystal formation, increasing toughness, a trick that modern metallurgy still struggles to replicate efficiently. Evolution's laboratory, operating over eons, perfected what humans only imitate in factories. The urchin's spines, too, are dynamic tools of sensory feedback. At their base, nerve clusters detect touch, pressure, and even chemical gradients in the water. Though it lacks a central brain, the urchin coordinates these signals through a nerve ring encircling its mouth. The result is distributed intelligence, a decentralized system that allows the creature to react swiftly to threats, align its spines toward motion, or adjust position to optimize feeding. In this way, the urchin feels the world not from a head, but from its entire body. Its awareness is diffuse, tactile, and ancient, a mode of perception that predates the vertebrate brain. Even reproduction in the urchin reveals the elegance of chemistry. During spawning, males and females release clouds of gametes into the water column, their synchronization triggered by temperature and moonlight cycles. The eggs are coated with chemical attractants that guide sperm through the currents, a microscopic navigation system built on molecular recognition. After fertilization, the embryos develop into free-swimming larvae, transparent and delicate, drifting with plankton until they settle and metamorphose. Within weeks, tiny spines begin to grow, and the first pedicellariae appear, microscopic jaws ready to defend a creature barely visible to the naked eye. Defense begins before awareness, coded into every stage of life. At night, under the faint shimmer of bioluminescent plankton, urchins take on an eerie beauty. Their spines catch stray photons, their bodies glowing faintly as they move across the substrate. Small shrimp hide among the spines, seeking protection from larger predators, a symbiosis born from trust in the fortress. Even here, the urchin's weapons shape relationships, providing shelter as much as deterrence. Evolution has turned hostility into habitat, defense into coexistence. The sea urchin's chemistry, like all great evolutionary inventions, serves balance rather than aggression. Its toxins do not hunt, they warn. Its spines do not pursue, they protect. Its micro jaws, tireless and precise, sustain both cleanliness and survival. In the grand equation of the reef, the urchin is both sentinel and sculptor, maintaining the fragile architecture of life that surrounds it. As dawn returns, waves wash over the reef, and the tide rises once more. The urchin remains unmoved, its spines shifting subtly to face the light. Around it, the ocean breathes. Predators glide by, currents change, sand settles, and still it endures. Every broken spine will be replaced, every threat countered by chemistry and form. For millions of years, this slow architect has survived the sea's endless cycle of creation and destruction. Its defense is not rage or speed, but patience reinforced by molecules and minerals. In a world built on motion, the sea urchin's power lies in staying perfectly, defiantly still. Morning sunlight filters through the surface of a shallow lagoon, scattering bands of gold across rippled sand. The water is warm and silent, broken only by the faint clicks of shrimp and the rustle of seagrass. From above, the scene seems tranquil, peace suspended in turquoise. But beneath the calm, something stirs, barely perceptible. A subtle mound of sand shifts. Two eyes, set wide apart, emerge from the seabed, followed by the slow undulation of a disc-shaped body. The stingray has been here all along, waiting, not for prey, not for conflict, but for safety. The stingray's body is a study in understatement. Its flattened shape allows it to merge seamlessly with the ocean floor, its skin speckled with fine patterns that mimic the patchwork of sand and shadow. Its pectoral fins, broad, wing-like expansions of muscle, beat gently as it glides through the water, leaving behind nothing but a whisper of disturbed sediment. Every motion is deliberate, every pause a calculation. For a creature so often misrepresented as dangerous, the stingray's true genius lies not in its sting, but in its restraint. The danger, however, is real and highly refined. Along the upper side of the tail lies its weapon, a barbed spine sheathed in a thin layer of skin loaded with venom glands. The spine itself is made of dentine, the same material that forms the core of human teeth, and it tapers into serrations that face backward, ensuring that once embedded, it resists removal. When a threat presses too close, a shark, a crocodile, or the careless step of a human, the ray lashes its tail in a whip-like arc. The epidermal sheath tears open, releasing venom into the wound through capillary action. The chemistry that follows is a storm of pain and confusion. The venom of a stingray is not designed to kill, but to repel. It contains serotonin, which amplifies pain by exciting nerve endings, and metalloproteases, enzymes that break down tissue and blood vessel walls, causing rapid swelling and bleeding. Catecholamines, similar to those produced during human stress responses, induce shock and drop blood pressure. Together, these compounds transform a single puncture into an overwhelming sensory assault. The predator, or intruder, withdraws, the ray escapes, and the lesson is remembered. In evolutionary logic, that is victory enough. Despite its power, the stingray's weapon is defensive, not predatory. Rays do not hunt with their tails, they hunt with silence. Their true strength lies in stealth and precision. Using electroreception, tiny organs called ampullae of Lorenzini scattered around their snouts, they detect the faint electrical signals emitted by the muscles of hidden prey. A buried crab's heartbeat, the minute contractions of a fish's gills, even the electrical whisper of movement across sand, all are visible to the ray in an invisible spectrum of current and charge. Once located, the ray settles over the prey, flaring its fins to trap it beneath, and then delivers a quick suction strike with its mouth on the underside. The strike is so fast and exact that the prey is engulfed before the sand above it settles. The venom is not for this purpose, it is a shield against interruption. For much of its life, the stingray is a passive resident of the shallows. It feeds on mollusks, worms, and crustaceans, crushing shells with plate-like teeth that grind as efficiently as millstones. It serves as a recycler in the benthic ecosystem, stirring sediment, aerating the seabed, and exposing buried nutrients that feed microscopic life. The act of feeding becomes ecological engineering, shaping the micro-landscape of the sea floor. Small fish often trail behind rays, darting in to snatch whatever fragments are left behind, a subtle but constant reminder that no act in the ocean exists in isolation. Still, the stingray's reputation overshadows its nature. It is often portrayed as an aggressor, a lurking threat. In truth, the stingray's sting is an act of desperation. Most incidents with humans occur when the animal is accidentally stepped on, triggering an instinctive reflex that evolved to save its life, not take another. Once the threat has passed, the ray glides away, leaving behind a wound that owes its severity not to aggression, but to molecular design. For the creature, it was merely survival in an unforgiving world. Under the microscope, the stingray's spine tells a story of evolutionary precision. Layers of dentine form concentric rings around a central pulp cavity, strengthening the structure while maintaining flexibility. The venom glands line the grooves along the spine, secreting their compounds into the protective sheath that encases it. This sheath is not a permanent fixture. It regenerates after each strike, rebuilding the weapon's chemical coating through continuous secretion. Even the process of replacing a lost spine, which can happen through breakage or natural shedding, is automatic. Within months, a new spine grows from the tail's dermal base, ready to assume its role. The cycle of armament is perpetual, and it reflects one of evolution's simplest truths. In the ocean, every advantage must be renewable. The effectiveness of the stingray's venom has driven centuries of fascination and fear. Indigenous coastal cultures across the Pacific and Caribbean long understood both its danger and its potential. Some use diluted venom in traditional medicine for pain management, recognizing its paradoxical relationship with the nervous system. How the same molecules that cause agony could, in small doses, numb it. Others crafted arrowheads from dried spines, preserving the natural serrations as barbs. In every case, the human relationship with this animal has been shaped by respect for its chemistry. The stingray was never hunted for sport, rarely eaten, and almost universally approached with caution. Modern medicine, too, has learned from this biochemistry. The standard treatment for a stingray wound involves immersion in hot water, sometimes nearing the limit of tolerable heat. The reason is simple and elegant. Most of the venom's proteins are heat sensitive. Exposure to temperatures above 40 degrees Celsius denatures the toxins, unraveling their molecular structure and rendering them harmless. The method requires no antidote, only knowledge of protein chemistry. It is a rare case where understanding the molecule provides immediate relief from its effect. Pain management and wound care follow, but the most critical step is as old as the sting itself: heat to undo what chemistry began. The stingray's anatomy is also a masterclass in energy economy. Its flattened body increases surface area, allowing it to rest on sand with minimal buoyant effort. Its gills, located on the underside, draw water through spiracles, small openings behind the eyes, rather than directly from the sediment, preventing clogging. This adaptation allows the stingray to remain buried for hours without surfacing, conserving energy while remaining perfectly hidden. When danger passes overhead, the only sign of life is the brief puff of sand as it exhales through its gill slits. Evolution has turned stillness into invisibility. Among stingrays, variation is as wide as the oceans they inhabit. The blue-spotted ribbon-tail of the Indo-Pacific flaunts iridescent markings that warn of its venom. The southern stingray of the Caribbean blends into sand with flawless precision. The massive short-tailed stingray of southern waters, weighing hundreds of kilograms, wields a spine the size of a dagger. Yet across species, the rule remains constant. Venom is not a sword, but a shield. Even the majestic manta ray, a relative without a venomous tail, carries the same lineage, a body shaped by the same evolutionary pressures, where the sting once served as defense before flight replaced fight. Viewed from above, a resting stingray is almost invisible, an imprint of geometry in the sand. Only the rhythmic rise and fall of its breathing betrays it. Beneath the surface, the tail lies coiled, the spine sheathed, the venom inert. It will remain that way for days, sometimes weeks, unless provoked. In that restraint lies a quiet intelligence. The stingray knows that the ocean rewards patience more than conflict. It endures by avoiding attention, not by seeking it. And yet in that quiet endurance, the stingray has become a symbol of balance in marine design, beautiful but armed, graceful but guarded. Its spine is not a weapon of war but of equilibrium, a tool that maintains the boundary between predator and prey. It is an expression of the ocean's oldest rule: survive without waste. As dusk settles over the shallows, the ray stirs again. A faint tremor of muscles lifts it from the sand. Its wings ripple once, twice, and it drifts into deeper water, the long tail trailing like a ribbon in twilight. Small fish scatter in its wake, and soon it disappears into the blue, leaving behind only the faint memory of movement across the seafloor. The reef returns to silence, unaware that one of its quietest inhabitants carries a weapon that can humble even the fiercest hunters. The stingray's legacy is not its sting, but its restraint, the marriage of elegance and defense perfected by time. Beneath its smooth skin lies a chemistry of consequence, a balance between pain and peace. It reminds us that in the ocean's endless experiment, the most enduring survivors are not those who strike first, but those who strike only when they must. In the dim light beneath a coral ledge, motion slows to a whisper. Sand drifts lazily in the current, particles suspended in beams of fractured sunlight that filter down through the turquoise water. Then, almost imperceptibly, the landscape shifts. What looked like a patch of mottled rock ripples and changes color, its texture morphing from coarse coral to smooth flesh. Two eyes, intelligent and unblinking, emerge from the camouflage. From beneath the coral, an octopus begins to move. An engineer, a chemist, and a hunter, wrapped in the same soft body. The octopus is a creature of paradox. It has no skeleton, yet it moves with precision. It has no voice, yet its body speaks in colors. It has no tools, yet it wields one of nature's most sophisticated chemical arsenals. Hidden inside its mouth, behind the beak that glints black like obsidian, lies a pair of venom glands linked to ducts that feed into a muscular organ, the salivary papilla. Through this papilla, the octopus can inject, exude, or apply its biochemical inventions with astonishing control. The weapon is liquid, not mechanical, and its delivery system as nuanced as any surgeon's hand. Underwater, the octopus's world is one of constant problem-solving. Prey does not come easily in the coral labyrinth. Crabs wedge themselves into crevices, clams lock their shells tight, and snails bury beneath sediment. But the octopus is a patient predator. It feels the world through arms lined with suckers, each sucker packed with tens of thousands of sensory neurons that detect texture, taste, and pressure all at once. It does not simply touch its environment, it analyzes it. When its suckers find the outline of a shell or the vibration of a hiding crustacean, the octopus moves closer, drawing the object toward its beak. What happens next is equal parts physiology and chemistry. The first step is mechanical. The beak, made of chitin hardened by cross-linked proteins, slices through shells and exoskeletons with ease. But when physical force is not enough, chemistry takes over. From ducts within the salivary glands, the octopus releases a potent mixture of enzymes and neurotoxins that travel through the papilla, a needle-like projection capable of piercing the smallest cracks. The saliva is a biological solvent, rich with proteases, lipases, and cephalotoxins. Together they begin to digest prey from within its own shell, liquefying muscle tissue and disabling movement. For a crab, the venom acts on sodium channels in the nerves, blocking transmission and inducing paralysis. For mollusks, specialized enzymes soften the ligament that holds the shell closed, allowing the octopus to pry it open effortlessly. Different species fine-tune this chemistry to their habitat and diet. The common reef octopus uses a balanced venom, strong enough to subdue but not destroy, preserving soft tissues for feeding. The Atlantic longarm octopus, which hunts polychaete worms, produces a lighter neurotoxin suited to smaller prey. But at the top of this evolutionary ladder sits the infamous blue-ringed octopus, a hand-sized creature that glows with iridescent warnings when provoked. Its saliva contains tetrodotoxin, a compound so lethal that a single gram could kill dozens of humans. This toxin, like that of the pufferfish, is not made by the octopus itself, but by symbiotic bacteria that live in its salivary glands. The relationship is ancient, a microbial alliance forged by evolution's quiet experiments. The bacteria gain habitat and nutrients, the host gains an unrivaled defense. When injected, tetrodotoxin binds to voltage-gated sodium channels in nerve membranes, blocking the flow of signals from brain to muscle. The result is total paralysis without loss of consciousness. Victims, whether fish or human, feel themselves suffocating while their minds remain fully aware, a terrifying testament to the power of molecular precision. Yet for the blue-ringed octopus, this chemistry is neither cruelty nor malice, it is necessity. Living in tide pools and shallow reefs, where escape routes vanish with the retreating tide, it cannot rely on speed or size. Its survival depends on deterrence. The flashing blue rings, produced by chromatophores and reflective iridophores under neural control, are a warning, evolved from experience. Touch me, and chemistry will finish what color began. But for most octopuses, venom is not the story of defense. It is the story of problem solving. Researchers have documented octopuses modulating their venom dosage depending on the prey's type and resistance. A crab with thick armor might receive a full injection, a shrimp merely a partial dose. This suggests an element of control, a learned association between prey behavior and chemical potency. In laboratory studies, octopuses quickly adjust their strategy when faced with new prey species, learning which tactics, drilling, suffocation, or venom injection, yield the best results. The connection between brain and biochemistry is direct. Intelligence dictates chemistry, and chemistry in turn sustains intelligence. Inside the octopus's brain, arguably the most complex among invertebrates, chemical learning manifests as behavioral flexibility. The central brain coordinates vision and planning, while the arms operate semi-independently, processing local sensory data and responding to stimuli in real time. Each arm is capable of autonomous motion, memory, and decision-making. When an octopus hunts, its body functions as a decentralized laboratory, the brain directing the experiment, the arms performing it, the glands supplying the reagents. The coordination between nervous system and venom system is so integrated that one might say the octopus does not use venom, it thinks with it. On coral reefs, this chemistry becomes choreography. An octopus drifts across a coral head, shifting from ochre to ivory to deep red, matching the textures of sponges and stones as though painting itself into the reef. A crab senses movement, freezes, then scuttles backward toward its burrow. The octopus hesitates, then pounces. Its arms close in a whirl of motion, suckers gripping rock and prey alike. The crab thrashes, claws striking wildly, but a single prick from the salivary papilla ends the struggle. The venom takes effect almost instantly. The crab's muscles seize, its legs stiffen, and within seconds it is motionless. The octopus does not devour it whole. Instead, it inserts its beak, injecting digestive enzymes that turn the interior into soup. In minutes, the shell is empty, a hollow trophy left on the sand. The mechanics of venom delivery are as fascinating as the chemistry itself. The salivary papilla is flexible, capable of drilling minute holes in shell surfaces through rotational abrasion. In some species, it is reinforced by mineralized rings, turning it into a biological drill bit. As the papilla penetrates, the surrounding tissue secretes a viscous fluid that prevents the venom from diffusing too rapidly, ensuring the full effect remains localized. This precision means that the octopus rarely wastes toxin. Each drop is calculated for maximum efficiency, a balance between energy investment and reward. Even in death, the octopus's chemistry continues to intrigue. Its venom has inspired pharmacological research into novel painkillers and cardiac treatments. Some peptides in cephalotoxin mixtures modulate ion channels with such specificity that they could one day be harnessed to treat epilepsy or neuropathic pain. The very molecule that silences prey could, in human hands, become a therapy that quiets disease. Nature's poisons, when understood, often become medicine. In ecology, the octopus's role as a venomous predator shapes communities more subtly than its speed or intelligence ever could. By targeting crustaceans and mollusks, it helps regulate populations that would otherwise overgraze reefs or smother coral. Its abandoned shells provide shelter for small fish and hermit crabs. Its behavior, hunting, hiding, discarding, creates microhabitats, turning the act of feeding into ecosystem architecture. Predators like moray eels, groupers, and sharks learn to approach octopuses with caution. The ripple effects of its venom extend outward through the food web, influencing behavior, distribution, and survival across species lines. Yet perhaps the most striking dimension of the octopus's venom is its connection to cognition. Unlike most animals that evolve toxins purely for defense or predation, the octopus integrates its chemistry into its decision-making repertoire. It experiments. Observers have noted octopuses testing new prey by touch before committing to a strike, or even using the tip of an arm to deliver a light dose, paralyzing without killing, allowing them to study the prey's response. This blend of curiosity and control reveals something profound, an awareness, however primitive, of cause and effect. In the octopus, intelligence and poison are not opposites, they are partners. As daylight fades, the octopus retreats into its den, rearranging stones at the entrance with deliberate care. Its arms tuck inward, skin fading from the colors of coral to the dull beige of sand. Inside, the creature rests, its metabolism slowing. The glands that brood venom all day now lie dormant, their secretions stored for the next hunt. Around the den, discarded shells and bones mark its domain, a silent record of chemical warfare waged not out of cruelty, but out of necessity. The ocean outside continues its rhythm. Waves break on the reef crest, plankton drift in moonlit swirls, and predators roam the dark. But within its hidden cave, the octopus embodies a truth older than the coral itself, that intelligence in nature is rarely divorced from danger. The same mind that solves puzzles also measures doses. The same arms that explore also deliver venom. It is a creature that survives through chemistry as much as thought, a being that blurs the line between brain and laboratory. In this balance between intellect and toxin lies the octopus's enduring enigma. It is both scientist and assassin, problem solver and predator. Its salivary syringes are not instruments of malice but of adaptation, a molecular conversation between evolution and ingenuity. And as it sleeps beneath the coral, surrounded by the quiet hum of its own chemical mastery, one thing becomes clear. In the deep, survival favors not the strongest or the fastest, but the cleverest chemist of them all. Dawn breaks across a shallow bay in the Indo-Pacific, and the water glows with subdued amber light. Below the surface, a field of seagrass sways rhythmically with the current. The silence is deceptive, a fragile calm in a world dense with communication. Flashes of color, waves of light, and chemical messages drifting unseen through the water. Here, among the slender blades, a cuttlefish lies half buried in sand, its skin a perfect mimic of the shifting seafloor. Patterns ripple across its body like wind over dunes, chromatic illusions driven by thousands of pigment cells under neural control. To most predators, it is invisible. But the cuttlefish's genius is not only to hide, it is to manipulate what others see, smell, and even desire. A shadow slides over the sand, a hunting jack, scanning for breakfast. The cuttlefish remains still, its chromatophores, tiny sacs of colored pigment, flattening into pale uniformity. Beneath the surface, electric pulses race across its nerves, triggering rapid contractions that expand or contract those pigment sacs by fractions of a second. In an instant, the animal's pattern changes from speckled to smooth, from rock to ripple, aligning perfectly with the light scattered by the waves above. The jack circles once, twice, then turns away. The cuttlefish exhales slowly through its siphon, propelling a faint puff of sand into the water. Danger for now has passed. But the cuttlefish's most astonishing defense does not lie in its skin, it lies in its ink. When escape becomes impossible, this creature deploys a chemical illusion so advanced it interferes with the predator's senses themselves. Unlike the squid's dark smokescreen or the octopus's blunt burst of confusion, the cuttlefish's ink is an integrated system of optics and chemistry, a weapon that manipulates behavior, not just visibility. It is both a visual barrier and a biochemical signal, designed to rewrite what the enemy perceives. The ink is stored in two specialized glands, one producing melanin, the dense pigment that forms its color, and another secreting mucus, amino acids, and dopamine-based compounds that alter its consistency and effects. When mixed and expelled through the siphon, this combination forms a rich, cohesive cloud that hangs in the water column like a drifting storm. The melanin particles absorb light, obscuring vision, while the mucus slows dispersion, allowing the ink to retain shape. In moments of desperation, the cuttlefish expels not a single dark cloud, but multiple pseudomorphs, dense ink masses roughly the size and shape of its own body. These phantom doubles drift in the current, mimicking the outline and texture of the animal itself. To a predator's eye, they are perfect decoys. The science behind this illusion is extraordinary. Melanin's optical properties scatter light across the visible spectrum, creating opacity in even minimal concentrations. But the cuttlefish's mucus modifies the ink's hydrodynamics, keeping it cohesive enough to appear as a distinct entity. This is not random secretion, it is calculated engineering, a composite material built for deception. While the predator lunges toward one of the false bodies, the real cuttlefish slips away, undetected, leaving behind a biochemical hologram that fades slowly into the current. It is an act of self-erasure, performed in seconds, yet the ink's most remarkable property is invisible. Beyond its visual function, it is laced with compounds that affect the predator's physiology. Dopamine, the same neurotransmitter that modulates mood and reward in vertebrates, acts as a behavioral suppressant in many fish species. When predators inhale the ink through their gills or taste it on the water, the dopamine can dull their appetite and reduce aggression. In laboratory tests, fish exposed to cuttlefish ink hesitate before striking again, as if momentarily disoriented or sated. This effect transforms the ink from simple camouflage into a biochemical deterrent, an agent that changes minds, not just sightlines. The predator loses focus, the chase dissolves, and the cuttlefish vanishes into safety. It is chemical persuasion at its most elegant. While its defensive tactics are legendary, the cuttlefish's use of chemistry extends far beyond escape. Inside its salivary glands, enzymes and mild neurotoxins target crustaceans and mollusks. During hunting, the cuttlefish stalks with stealth and patience, its body undulating gently in synchrony with the waves. Once within range, it flashes a hypnotic display of oscillating light and shadow along its arms. This flickering pattern disorients small prey, freezing them in momentary paralysis, a visual trick of the nervous system. The cuttlefish then extends two tentacles with lightning speed, grasping the prey and injecting saliva that immobilizes it further. Within moments, the meal is subdued. What began as color becomes chemistry. What began as motion ends in molecular control. The coordination between its brain, skin, and glands borders on artistry. The cuttlefish's nervous system is divided between a large central brain and a network of peripheral neurons distributed through its arms and skin. Each chromatophore is connected to its own nerve ending, meaning the animal can control individual color cells in patterns that ripple like digital code across its body. This neural precision allows not only camouflage but communication, bursts of stripes and flashes that signal aggression, submission, or courtship. When coupled with chemical releases from its skin and ink glands, these displays become multisensory broadcasts, messages that operate on sight, scent, and behavior simultaneously. To the cuttlefish, the ocean is a field of communication channels, and it has mastered every one. But even perfection carries risk. The act of inking is costly. Producing melanin and mucus requires energy and nutrients that cannot be replaced quickly. Frequent use can leave the animal vulnerable for days, unable to defend itself until the glands refill. For this reason, cuttlefish deploy their ink only when truly necessary. Most rely instead on their unmatched camouflage, changing not just color but texture by raising tiny muscular bumps called papillae across their skin. In a fraction of a second, smooth flesh becomes rough coral, flat sand, or tangled algae. This is not mimicry, it is simulation. Their control of light, shape, and shadow creates a level of invisibility that even cameras struggle to capture. When hiding fails and escape begins, the ink becomes their final word. It is deployed in patterns as strategic as military countermeasures. In one burst, the ink cloud obscures pursuit. In the next, pseudomorphs draw attention away. If escape requires sprinting, the cuttlefish releases lighter, quickly dispersing ink that vanishes faster, creating confusion rather than decoys. The animal calibrates its chemistry based on situation and threat, a reactive intelligence encoded not in thought, but in evolution's refinement. In essence, it has learned to weaponize perception itself. This dual use of optical and chemical strategies has drawn scientists to study the cuttlefish as a model for artificial intelligence and material science. Engineers analyze its chromatophore networks to design adaptive camouflage fabrics that shift color in real time. Chemists examine its ink's adhesive properties, noting how the polymeric structure clings to surfaces underwater without dissolving, a potential template for bio-inspired glues. Even neuroscientists study its dopamine-laden ink for clues into cross-species neuromodulation. The cuttlefish, once dismissed as a curious cephalopod, now stands as a model for innovation in human technology. Yet for all its intelligence, the cuttlefish remains vulnerable. Its lifespan is short, rarely more than 2 years. It lives quickly, grows fast, and invests heavily in reproduction. When mating season arrives, males compete not with violence but with display, waves of pulsating color racing across their bodies in a silent courtship performance. The ink glands, now repurposed, also release pheromonal compounds that guide potential partners through murky water. Even in love, chemistry reigns. Once eggs are laid and the female guards them, her life cycle nears its end. The ink that once defended her will soon diffuse one last time into the sea, absorbed and forgotten. At dusk, as the tide retreats, a cuttlefish glides over a coral ridge, bathed in the fading glow of sunlight. A barracuda approaches, drawn by movement. For an instant, predator and prey lock eyes. Then, in a pulse of motion, the cuttlefish releases a perfect black sphere into the water, dense, opaque, and alive with swirling reflections. The barracuda strikes, teeth closing on emptiness. By the time the ink cloud drifts apart, the cuttlefish is gone, vanished into the background. What lingers is not a chase, but a question. Did it disappear, or did it make you believe it had? The cuttlefish's ink is more than defense, it is narrative control. It edits perception, scrambles intention, and rewrites the chemistry of behavior. Within a single jet of darkness lies a fusion of physics, neurology, and biochemistry, a masterpiece of evolution that proves survival is not always about strength or speed, but about understanding how others see and think. In the theater of the ocean, the cuttlefish is both magician and scientist, performing acts of illusion with precision that borders on art. Its ink is not a curtain to hide behind. It is a story told in molecules, one that ends only when the water clears and the world once again believes its own eyes. In the soft, dappled light of a coral reef, the water seems peaceful. Slow waves drift between limestone towers, scattering rainbows of light across a mosaic of colors. Small damselfish dart through coral branches, shrimp click in staccato bursts, and plankton shimmer in the sunlit haze. Yet, nestled between those vibrant colonies lies a creature that defies its stillness. It does not swim, it does not chase, and it does not flee. But within its body waits an arsenal so vast that few predators dare to test it. The sea anemone, delicate in appearance and rooted like a flower, is a living minefield, an organism that has weaponized chemistry, physics, and patience. From a distance, it looks harmless, a crown of soft tentacles swaying gently with the current, each filament tracing lazy arcs through the water. But every one of those tentacles is an ambush in waiting. Inside them are millions of tiny capsules, nematocysts, that function as microscopic harpoons. Each one is coiled under immense internal pressure, ready to fire in less than a millionth of a second when triggered by touch or chemical cues. To the human eye, the strike is invisible. To a planktonic crustacean brushing too close, it is instantaneous death. The harpoon pierces skin, anchors, and injects venom all in one fluid motion. What appears as motionless beauty is, in truth, a hair-trigger predator armed with a cellular weapon unmatched in nature's speed and precision. The firing of a nematocyst is one of biology's most violent events at a microscopic scale. Inside the capsule, osmotic pressure builds to several times that of a car tire, maintained by ions trapped in solution. When sensory cilia on the tentacle surface detect contact, a molecular latch releases and the capsule explodes. A coiled tubule unravels outward at speeds exceeding 30 meters per second, reversing itself like a sock turning inside out. The tip of this thread, barbed and razor-sharp, punctures the prey's surface, delivering a pulse of venom into its cells. It is mechanical engineering perfected by evolution, an organic spring powered by chemistry, honed through hundreds of millions of years of selective pressure. The venom itself is a complex cocktail of neurotoxins, pore-forming proteins, and enzymes that destroy membranes and hijack signaling pathways. The neuroactive peptides target ion channels in nerves, freezing muscles mid-motion, while cytolysins punch holes in cell membranes, allowing calcium ions to flood in and rupture the victim's tissues. The prey does not die from trauma, but from chaos. Its cells literally short-circuit. Within seconds, the paralyzed organism is reeled inward by coordinated contractions of the tentacles and swallowed whole into the anemone's central cavity, where digestive enzymes complete the work chemistry began. Despite its plant-like appearance, the anemone is a hunter, and a surprisingly dynamic one. It can contract its tentacles in coordinated waves, responding to both mechanical and chemical signals in the water. These reflexes are controlled not by a brain, but by a diffuse nerve net spread throughout its body. Every cell in this network communicates locally with its neighbors, creating patterns of response that ripple outward like choreographed thought. The result is a form of distributed intelligence, a living example of how even creatures without centralized nervous systems can process information and make decisions about the world around them. But perhaps the most remarkable use of chemistry in the sea anemone's arsenal is not aimed at prey, but at competitors. On crowded reefs where space is life, anemones wage silent wars through allelopathy, the release of chemical compounds that damage or kill neighboring organisms. When two anemones of different species share a rock, they establish invisible borders defined not by sight or sound, but by molecular warfare. Specialized acrohagi, bulbous outgrowths filled with stinging cells, extend toward rivals and secrete cytotoxic peptides that cause tissue necrosis on contact. Over time, these chemical exchanges carve territorial lines across the reef, delineating patches of dominance that can last for decades. The coral surface becomes a patchwork map of invisible aggression, each border patrolled by scent and molecule. Some species take this territoriality to astonishing extremes. The carpet anemone, Stichodactyla gigantea, uses allelopathic compounds to prevent the settlement of coral larvae nearby, ensuring its domain remains unchallenged. The chemicals leach slowly into the surrounding water, altering the substrate's chemical composition just enough to discourage colonization. In effect, the anemone engineers its own neighborhood, maintaining open real estate through chemical deterrence. Evolution has turned a stationary creature into a molecular strategist, capable of shaping the environment around it without ever moving an inch. Yet even this fortress of venom harbors partnerships. Within its toxic embrace live symbiotic allies, clownfish, shrimp, and crabs, that have evolved remarkable resistance to the sting. The clownfish, perhaps the most famous of these tenants, gains sanctuary among the anemone's tentacles. Its secret lies in chemistry. Over time, the fish coats itself with a layer of mucus rich in glycoproteins that mimic the anemone's own surface. To the nematocysts, the fish is chemically invisible. This disguise prevents the firing mechanism from activating, allowing the clownfish to live unharmed. In return, it cleans debris, provides nutrients through waste, and drives away small predators. What was once a minefield becomes a fortress shared through biochemical diplomacy. Other organisms exploit the anemone's defenses in more parasitic ways. Certain hermit crabs attach small anemones to their shells, transforming themselves into mobile fortresses. When threatened, the anemones flare their tentacles, creating a barrier of stings that deters would-be predators. In return, the anemone gains transport and access to food scraps from the crab's meals. Even sea slugs, notorious for repurposing other species' defenses, feed on anemones and store undischarged nematocysts within their own tissues, a theft of weapons at a cellular level. In the ocean, chemistry does not belong to the creature that makes it. It belongs to whoever can use it best. Beneath their outward stillness, anemones are dynamic organisms, constantly adjusting their internal balance to the shifting chemistry of their surroundings. Their tissues contain photosynthetic symbionts, zooxanthellae, that provide nutrients through the conversion of sunlight. This relationship, similar to that found in corals, allows the anemone to survive in nutrient-poor waters, blending animal predation with plant-like photosynthesis. When environmental stress, such as heat or pollution, disrupts this symbiosis, The anemone expels its symbionts in a process known as bleaching. Without them, its colors fade, and its energy reserves deplete. In these moments, the minefield weakens. Even the most fearsome chemical arsenal cannot defend against climate's slow assault. The sea anemone's capacity for regeneration adds yet another layer to its resilience. Torn tentacles regrow, damaged tissues seal within hours, and even split individuals can clone themselves into genetically identical copies. This ability allows anemones to colonize new areas and recover from injury, creating vast clonal colonies that dominate patches of reef. In such communities, each polyp is a separate individual, yet linked genetically and chemically, forming a single defensive network. The collective can respond to threats in unison, stinging, retracting, or expanding together. It is the ocean's version of a biological minefield grid, alive, coordinated, and self-repairing. To watch an anemone in slow motion is to see warfare disguised as grace. The tentacles undulate gently in the current, each movement masking lethal readiness. Tiny fish drift by, sometimes grazing against the tips without consequence, immune through evolved resistance or sheer luck. Others are not so fortunate. A small shrimp brushes too close, and within a blink it is ensnared. The tentacles tighten, the body folds inward, and within minutes the prey is gone. The reef resumes its tranquil rhythm, as if nothing happened. Yet in that quiet moment, the anemone has demonstrated the ruthless efficiency of a predator that does not need to move to kill. In the hierarchy of reef life, the sea anemone occupies an unusual position. Both predator and habitat, both destroyer and provider. Its toxins kill, yet they also protect. Its chemicals carve borders, yet they also foster partnerships. It blurs the line between animal and environment, its body serving as both organism and ecosystem. The minefield is alive, and its weapons are not merely tools of war, they are instruments of ecological order. As sunlight fades and the tide shifts, the anemone retracts slightly, its tentacles curling inward like closing flowers. The reef darkens, and bioluminescent plankton begin to sparkle in the water column. A nocturnal fish glides past, keeping a careful distance from the shadowed shapes that dot the seabed. The anemone waits in silence, every cell armed, every tentacle alive with potential. It has no eyes to see the danger it poses, no consciousness of its boundaries or its allies. Yet through chemistry alone, it commands respect from every creature that shares its reef. In a world defined by motion, the sea anemone thrives by doing nothing. It lets the ocean move around it, lets chemistry speak for it, and lets evolution refine its defense molecule by molecule. It is proof that survival in the sea does not always belong to the swift or the strong. Sometimes it belongs to the still, to the organism that turns stillness into strategy and patience into power. And so the anemone remains, a sentient minefield disguised as a flower, shaping life on the reef with invisible hands of venom and molecular diplomacy. Morning light ripples across the coral reef like liquid gold. Tiny particles suspended in the water scatter the sun into a slow shimmer, each beam catching scales, shells, and soft anemone tentacles swaying in rhythm with the current. Among the wavering forest of stinging arms, a flash of orange and white darts, a clownfish weaving confidently through what should be a lethal maze. Around it, the sea anemone's tentacles brush and curl, bristling with nematocysts that could paralyze small prey in a blink. Yet this little fish swims untouched, even resting between those venomous strands as if sleeping inside a bed of loaded weapons. To the reef's inhabitants, it seems an impossible alliance. To biology, it is a masterpiece of chemical deception. The clownfish's home is the sea anemone, an organism that lives by killing. Each tentacle contains batteries of coiled, stinging cells ready to fire at the slightest hint of alien chemistry or touch. These cells, nematocysts, are powered by osmotic pressure that hurls harpoon-like tubules with explosive force, injecting toxins designed to pierce, paralyze, and digest prey. But when the clownfish glides among them, nothing happens. The tentacles caress rather than sting, folding softly around the fish like living silk. This is not magic or luck, it is chemistry, honed by evolution to a near-perfect disguise. On the clownfish's skin lies a mucus layer, unlike that of any other reef fish. Most reef dwellers secrete mucus rich in proteins and lipids that mark them as foreign to the anemone's chemical sensors. But clownfish mucus is dominated by glycoproteins and amino sugars that mimic the anemone's own cellular coatings. To the tentacles, the clownfish's presence is not alien. It feels like part of the anemone itself. The nematocysts do not recognize a target. Their molecular triggers remain silent. In effect, the clownfish wears a biochemical mask, an armor of false identity that prevents the stings from firing or weakens their reaction to harmless contact. It is one of the ocean's most sophisticated acts of molecular espionage. Yet this protection is not instant. Juvenile clownfish must earn their disguise through a ritual scientists call the acclimation dance. When a young fish first approaches an anemone, it does so cautiously, its movements hesitant and deliberate. It circles the tentacles, darting forward and retreating in small, nervous bursts. Then, it begins to touch the anemone's tentacles with quick, tentative taps, retreating immediately each time a mild sting occurs. Over hours, sometimes days, it repeats this behavior, brushing its body against the tentacles for slightly longer intervals with each attempt. The purpose is dual: to coat itself with trace mucus from the anemone's surface, and to desensitize the host's nematocysts to its presence. The process is both chemical and behavioral, a conversation of molecules and instinct. By the end of this dance, the clownfish's mucus is layered with the anemone's biochemical cues, and the anemone's cells no longer view it as prey. The minefield has been disarmed, at least for one small intruder. The clownfish's mucus is more than camouflage. It is a living, dynamic interface. Embedded within it is a complex microbiome of bacteria, some inherited from parents, others acquired from the host anemone. These microbes are not passive passengers. Many play active roles in maintaining the fish's chemical disguise, producing enzymes that modify surface sugars or secrete compounds that inhibit microbial growth on both fish and anemone. In laboratory analyses, microbiologists have found striking overlaps between the bacterial communities on clownfish skin and the tissues of their host anemones. It is as if the two species share not only a home, but also a microbiological identity, bound by invisible symbionts that mediate peace at the cellular frontier. The benefits of this alliance extend in both directions. The clownfish gains sanctuary in one of the most secure fortresses on the reef. Predators, wrasses, groupers, and moray eels know the cost of approaching too closely. Even the shadow of a pursuing predator will cause the clownfish to dart between the tentacles, vanishing into safety. Meanwhile, the anemone reaps its own rewards. The clownfish is an active tenant. It cleans the anemone's surface by removing debris and parasites, and its constant motion improves water circulation around the tentacles, delivering more oxygen to the anemone's tissues. In return for protection, it offers maintenance. Moreover, the clownfish's waste provides nitrogenous nutrients, which fuel the growth of the anemone's photosynthetic symbionts, tiny dinoflagellates that live within its cells and generate energy from sunlight. In this small domestic exchange, fish and anemone sustain one another through chemistry and behavior, forming one of the ocean's most iconic examples of mutualism. At night, when the reef grows quiet, the relationship becomes even more intimate. The clownfish nestles deep between the anemone's tentacles, its body pressed close to the oral disc. Here, in the warmth of the anemone's pulsating tissue, it rests. Some species even exhibit nocturnal coordination with their hosts, synchronizing movement patterns in response to subtle shifts in current and temperature. This proximity is not without risk. Any lapse in chemical mimicry could provoke a lethal sting. Yet the clownfish trust the relationship completely, a testament to the evolutionary refinement of their shared biochemistry. Beneath the surface of this partnership lies a story of adaptation, shaped by evolutionary tension. Anemones are ancient creatures, some of the earliest predators on Earth, thriving long before fish evolved bones or scales. Over millions of years, as reef ecosystems diversified, a handful of small, colorful fish found ways to exploit these toxic guardians. The earliest ancestors of clownfish likely began by hovering at the edges of anemone colonies, feeding on leftover scraps and using the tentacles as partial shelter. Gradually, Individuals with thicker mucus layers or mutations that dampened nematocyst sensitivity survived longer. Over generations, biochemical mimicry deepened, transforming opportunism into symbiosis. Today's clownfish are the product of that long chemical negotiation, a lineage of trespassers turned tenants, tolerated by what was once an implacable killer. The partnership is not universal. Each species of clownfish tends to bond with specific anemone species, a pattern driven by compatibility between their mucus chemistry and the anemone's surface proteins. This selectivity reduces conflict but also creates dependency. Remove the anemone, and the clownfish becomes vulnerable, its bright colors a beacon to predators. Remove the clownfish, and the anemone loses its cleaning crew and its nutrient supply. The relationship is delicate, interwoven at every level from ecology to molecular signaling. It demonstrates how survival in coral ecosystems depends not on individual strength, but on interdependence, an economy of cooperation mediated by chemistry. The clownfish's bright coloration, often assumed to be a liability, is in fact a message of mutualism. The vivid orange and white bands advertise the anemone's presence, warning would-be intruders of its stinging defense. In a sense, the clownfish acts as a living signpost, broadcasting danger to the reef. The signal benefits both partners. Predators learn to associate those colors with the anemone's venom, and the fish can swim more boldly, secure in the reputation of its home. It is an evolutionary form of shared branding, a visual alliance written across skin and tentacle alike. Researchers have discovered even deeper subtleties in this chemical truce. The anemone's nematocysts are not permanently inactive around the clownfish. Instead, their firing threshold shifts. When the fish's mucus touches the tentacles, the sensors that detect non-self molecules register familiarity, reducing the probability of discharge. But if the clownfish is injured or its mucus disrupted, say by abrasion, disease, or stress, the protection falters. Sudden stings can occur, a reminder that the alliance is conditional and must be chemically maintained. To live within the minefield, the clownfish must constantly renew its disguise, replenishing mucus and grooming itself against the anemone's surface. The relationship is not peace by forgiveness, it is peace by ongoing negotiation. The ecological consequences of this partnership reach beyond its participants. The activity of clownfish around their hosts influences nutrient cycling and local biodiversity. By scaring away herbivorous fish that might damage the anemone and attracting plankton through waste and movement, clownfish alter microhabitat composition. Some scientists refer to them as reef gardeners, their presence subtly reshaping the chemical and biological landscape around anemone colonies. Even their sound, gentle popping noises used in communication, has been shown to affect plankton behavior nearby. In the reef's intricate symphony, these small fish play notes far beyond their size. As evening settles over the coral garden, light fades to blue and the current slows. The clownfish retreat deeper into their hosts, vanishing among the undulating tentacles that close protectively around them. The anemone glows faintly in the dim light, its tissue alive with the pulse of symbiotic algae. Together, fish and anemone form a single living unit, a predator fused with its prey, a shelter fused with its tenant, an ecosystem contained within a handshake of molecules. The reef drifts into stillness, but the lesson endures. Survival in the ocean is rarely about brute force. More often, it is about chemistry, the invisible dialogues that turn danger into cooperation and enemies into allies. The clownfish does not conquer its deadly host, it convinces it. Through sugars, proteins, and bacteria, it rewrites the rules of recognition, teaching a killer to accept a guest. In a world where every encounter is a potential war, the clownfish thrives by mastering diplomacy at the molecular level. And so, as the night deepens, the clownfish sleeps unharmed among weapons designed to destroy it. A creature that has not diffused the minefield, but learned to sing its chemical password. Evolution, in this small corner of the reef, found a way to turn poison into peace. The tide is retreating, and the reef awakens in the light of low morning. Shallow pools left by the waves glimmer like mirrors, each one a microcosm, urchins wedged under rocks, gobies darting through coral rubble, snails inching across sand grains. Among them scuttles a figure both bold and deceptive. Its carapace is painted in impossible colors, crimson and gold, with streaks of violet and jet. Sunlight flashes across its claws, and its eyes glint like wet obsidian. To the casual diver, it looks like a jewel crawling over coral. But beneath that armor lies one of the most dangerous chemical arsenals in the ocean. This is a xanthid crab, a creature that does not manufacture its own poisons, but carries within it the chemistry of others, a living vessel of borrowed toxins. The reef has always been a laboratory of chemical warfare. Corals build acids to shape rock, sponges brew antifouling compounds, cone snails wield molecular syringes. Yet few creatures illustrate the evolutionary art of chemical appropriation quite like these crabs. They are small, rarely larger than a human thumb, and slow by crustacean standards. Many species lack the heavy claws or quick reflexes that typify predatory crabs. Instead, they wear warning colors, bright yellows, stark blacks, and vivid oranges that stand out against the dull greens of the coral bed. In a world where blending in is survival, these crabs do the opposite. They flaunt visibility as a threat, declaring to the reef, "I am dangerous to touch." And they are. Inside their tissues circulate some of the most potent neurotoxins known, tetrodotoxin and saxitoxin, compounds so lethal that a few milligrams could stop a human heart. These are the same molecules that make pufferfish deadly and render certain shellfish unsafe during red tides. Yet the crabs themselves are not victims of their own armament. Evolution has rewritten the locks on their nerves. Their sodium channels, the microscopic gates that allow nerve impulses to fire, carry structural mutations that prevent the toxins from binding. The molecule that would silence a mussel or paralyze a predator slides harmlessly past, unable to grip its target. In this immunity lies the key to their survival, for these crabs do not produce their poisons, they collect them. The process begins invisibly, with diet. Xanthid crabs graze on a toxic buffet, blue-green algae, cyanobacteria, and small invertebrates that host toxin-producing symbionts. Some species maintain permanent bacterial colonies in their gut or glands, microbial partners that continuously generate tetrodotoxin and saxitoxin. From there, the crabs accumulate and store the molecules throughout their tissues, especially in the hepatopancreas, the organ that functions as both liver and stomach. Unlike vertebrates, which would quickly succumb to such poisons, these crabs have evolved precise transport mechanisms that isolate the toxins from sensitive tissues while allowing their concentration to build elsewhere. Over time, the entire crab becomes a chemical reservoir. Every muscle, every claw, every fiber of its flesh holds traces of molecular death. Biologists studying these relationships discovered something remarkable. The crabs are not passive hosts, but active curators of their toxicity. When reared in laboratory aquariums and fed toxin-free diets, they gradually lose their lethality. The bacterial symbionts dwindle and the crab's tissues become safe to consume. Reintroduce them to their natural toxic food sources and the poison returns. This dynamic reveals the ecological truth of their power. Toxicity is not a trait but a collaboration. It is the result of a symbiosis between crab and microbe, predator and molecule, evolution and opportunity. For predators, a single mistake is often fatal. A moray eel that snaps at a xanthid crab may live long enough to regret it. The toxins act with ruthless efficiency, blocking sodium channels in nerve and muscle cells, halting the transmission of electrical signals. The heart falters, the diaphragm stiffens, and within minutes, paralysis spreads through the body. There is no pain, only silence, nerves frozen mid-command. Few creatures make that mistake twice. In this way, the crab's toxins radiate influence far beyond its own survival. They teach the reef's hunters caution, reshaping the behavior of entire food webs. Chemical memory becomes an evolutionary teacher. But the crab's chemistry is not just for defense. It also shapes its ecology. On reefs where these toxic species thrive, fewer fish graze on small crustaceans, shifting predation pressure toward non-toxic species. The result is a subtle redistribution of energy and behavior. Some cleaner fish learn to avoid the bright warning patterns entirely. Others evolve to tolerate mild versions of the same compounds, an evolutionary negotiation that plays out molecule by molecule. In the microcosm of coral ecosystems, every toxin is a political tool, every defense a statement that ripples outward. Contrast this with the more common, harmless crabs that share their environment. Many rely not on borrowed chemistry but on foul-tasting secretions, bitter compounds excreted from specialized glands that discourage predators without risking lethality. Others coat themselves with sponges or algae that emit repellent odors, transforming camouflage into chemical disguise. Still others wield behavior instead of venom, raising claws in threat postures, waving limbs in rhythmic displays, or using decoys of sand to confuse attackers. Against this spectrum of defense, the xanthid's approach is extreme, a gamble that trades speed, armor, and camouflage for biochemical certainty. To the human eye, these gaudy crabs seem almost ornamental. Their patterns, neon stripes, polka dots, and iridescent sheens, evoke the extravagance of tropical birds or poison dart frogs. Yet this beauty is evolutionary propaganda, a language shared across phyla. A predator that bites such brilliance does not survive to forget it. In this sense, the crab's colors are not decoration but communication, a billboard advertising chemical danger. In the language of survival, brightness is honesty. The reef, for all its chaos, runs on rules that even its smallest inhabitants obey. The source of the toxins themselves traces back not to animals, but to the invisible world of microbes. Cyanobacteria drifting in the water column and symbiotic bacteria residing in sediments synthesize tetrodotoxin and saxitoxin as part of their own defense systems. These compounds are molecular masterpieces, small, stable, and deadly efficient. They have evolved not to kill indiscriminately, but to deter grazing by microscopic predators. In the ocean's endless recycling of chemistry, these bacterial molecules pass upward through the food web, accumulating in larger organisms. The xanthid crabs stand at a crossroads in that transfer, turning microbial warfare into macro-scale deterrence. The mechanisms of storage within the crab's tissues are equally fascinating. Specialized binding proteins latch onto the toxin molecules, preventing them from floating freely in the bloodstream where they could interfere with the crab's own nerves. Instead, they are sequestered into vesicles, tiny cellular compartments that hold the toxins safely until a predator attempts a bite. When tissue is torn, the molecules are released directly into the attacker's mouth, bloodstream, or gills. The poison is not delivered by fang or spine, it is delivered by consumption itself. The crab's body is both bait and weapon, and the act of eating it completes the trap. The evolutionary implications stretch further. Similar adaptations appear in other marine animals, pufferfish, ribbon worms, certain starfish, all of which share the same microbial partners. The convergence suggests that in the ocean, chemistry travels faster than genes. Organisms across distant lineages have learned to outsource defense to the same molecular producers. The xanthid crab is thus not merely toxic, it is a biological courier in a vast network of chemical alliances. Humans too have encountered these crabs, often to their peril. In coastal regions of the Indo-Pacific, where small reef crabs are sometimes collected for food, xanthid species have caused fatal poisonings, indistinguishable from pufferfish intoxication. No amount of cooking destroys tetrodotoxin or saxitoxin. Both molecules are heat-stable and resist breakdown by digestive enzymes. The victim's symptoms mirror those of the crab's natural predators: numbness, paralysis, and respiratory arrest. Ironically, these creatures have never meant harm to humans. Their warning colors, visible even in shallow tide pools, were always meant to prevent, not provoke, consumption. Yet not all interactions between crab and chemistry are so grim. Scientists studying xanthid species are uncovering clues to new medical applications. Tetrodotoxin, despite its potency, has shown promise as a painkiller capable of blocking nerve signals without addiction or tolerance. In small controlled doses, it may one day relieve the very suffering it once caused. Evolution's poisons, understood correctly, often become medicine's tools. The xanthid crab, indifferent to its role, continues its patient work on the reef, embodying the duality that defines life beneath the sea, harm and healing entwined in a single molecule. As dusk falls, the reef glows under fading sunlight. The xanthid crab pauses in the shadow of a coral outcrop, its claws flexing lazily as plankton drift through the current. Around it, predators roam, wrasses and eels and small reef sharks, but none come close. Its bright shell gleams like a warning flare in the dim blue. In a world where camouflage rules survival, it thrives by breaking the rule entirely. The crab's safety lies not in hiding, but in being known for what it carries and what it can unleash. It does not need to run, it does not need to fight. The ocean remembers its lesson. To touch it is to risk silence, to eat it is to die. And so the crab remains, a slow-moving emblem of chemistry's power, proof that in the arms race of evolution, even the smallest creatures can borrow the deadliest weapons and turn survival into a masterpiece of molecular deceit. In the dim blue of a rocky reef, where light bends and fades into the green shadows of kelp, two lobsters approach one another. The water between them is still for a moment, thick with tension, vibrating faintly with the low hum of the current. Their antennae sway like whips, their claws poised but motionless. Then, before the first strike, one of them fires. Not a claw, but a cloud. A jet of water bursts from beneath its eyes, invisible except to the camera's slow-motion lens. It's not ordinary water, it's urine, laced with chemistry that carries identity, intention, and challenge. The ocean's oldest warriors begin their battle not with blows, but with scent. To most creatures, chemical communication is subtle, a whisper in the current. But for lobsters, it is a language written in molecules, transmitted through plumes of excreted fluid aimed with startling accuracy. Small nozzles at the base of their antennae act as precision jets, expelling pulses of urine that drift through the seawater like invisible smoke signals. Within those plumes are hundreds of compounds, peptides, amino acids, and metabolic byproducts that together form a chemical fingerprint as unique as a voice print in air. To a lobster's sensory world, every molecule tells a story. Who the other is, how large they are, whether they are a threat, a mate, or an intruder trespassing into private shelter. This chemical language replaces words with gradients and concentration. As the plume spreads, it is passed by specialized receptors on the opponent's antennae and mouthparts. Those sensory hairs detect specific molecular structures, each one triggering a cascade of neural signals that translate the chemical cloud into information. Within seconds, one lobster knows if the other is familiar or foreign, dominant or subordinate, male or female. The interaction that follows, whether a clash of claws or a graceful withdrawal, depends entirely on that first exchange of molecules. In this underwater world, chemistry decides destiny long before aggression is physical. When two males contest a burrow, the exchange unfolds like ritualized warfare. The larger lobster, often the territory holder, releases an initial plume, a territorial declaration that saturates the surrounding water. The smaller rival responds in kind, countering with his own chemical profile. The molecules intermix, and receptors interpret not only presence but proportion. The strength and composition of each scent reveal relative size, vigor, and recent victories. A dominant male's urine contains distinct peptides that mark his status, reinforced by hormonal cues linked to serotonin levels and muscular readiness. A subordinate, recognizing that molecular authority, will often retreat without a fight. In a sense, the battle is resolved by chemistry before claws ever touch. When conflict escalates, however, the exchange grows physical. Lobsters grapple with immense force, claws locking in combat that can last minutes. But even in the chaos of struggle, the chemistry continues. Each blow releases additional bursts of urine into the water, reinforcing identity and intent. In laboratory studies, when scientists blocked these urine jets, fights became longer and more violent, proof that chemical communication acts as a moderating force, a negotiator embedded in physiology. The molecules are not just signals, they are social contracts that govern behavior across the species. Outside of combat, the same language turns tender. During the mating season, females nearing molting release plumes of their own, rich with hormonal compounds that announce reproductive readiness. For a brief window after shedding their old shell, they are soft, vulnerable, and receptive. Their urine becomes an advertisement, a blend of pheromones that can travel several meters downstream. Males detect it instantly, their antennae vibrating as if sensing an electrical field. The response is immediate but restrained. The same male that once defended his den with violence now adopts a role of guardian, allowing the female to share his shelter while her new exoskeleton hardens. During this time, he releases his own plumes, chemical reassurances that deter rivals from approaching and signal his protective intent. The same organ that fired challenges now emits calm, defensive loyalty. Evolution, efficient as always, has repurposed aggression into romance. The precision of this signaling is astonishing. When visualized with fluorescent dye under controlled conditions, the urine jets form coherent streams that can reach several body lengths, arcing forward in tight beams before dispersing into turbulent swirls. The plume's shape depends on water current, jet angle, and the rhythmic contractions of bladder muscles beneath the lobster's carapace. Each pulse is timed, deliberate. In controlled environments, lobsters even aim differently depending on context. Direct streams for aggression, broader dispersal for courtship. The ocean may dilute scent quickly, but these crustaceans have evolved the mechanical finesse to make every molecule count. Inside the plume lies biochemical nuance. Researchers analyzing its contents have identified small peptide fragments likely derived from metabolic proteins, along with nitrogenous wastes, steroid hormones, and unique volatile compounds. Together, these molecules encode complex messages, part identification card, part emotional broadcast. Some compounds indicate recent feeding or stress, others correlate with the presence of reproductive hormones. The blend can change daily, reflecting diet, age, or social encounters. A lobster's chemical profile is therefore not static. It evolves, tracking its biography in scent. To neighboring lobsters, this profile provides more than simple identity. It conveys history. Chemical tagging extends beyond direct interaction. Dominant lobsters often mark their shelters by releasing urine inside crevices or along rock walls. The scent adheres to surfaces, lingering for hours or days depending on current and temperature. Intruders entering such marked territories encounter a faint but unmistakable warning, a chemical boundary invisible to the human eye. These odor borders function like the claw marks of a bear or the song of a bird. They delineate ownership and hierarchy without the need for confrontation. The ocean, rich with chemical gradients, becomes a living map of territories defined by invisible signatures. Such communication is not limited to adults. Even juvenile lobsters rely on chemical cues from conspecifics to navigate their early environments. Newly settled young detect the scent of adults through seawater, using it to locate potential shelters and gauge safe distances from predators. This sensitivity to chemistry begins early in life, long before claws or size offer protection. For them, survival is about reading the molecular mood of their surroundings, an instinct that will define every future interaction. The physiological machinery behind this system is as intricate as the behaviors it drives. The antennal glands, which serve as excretory organs, double as the control centers for chemical communication. They filter nitrogenous waste and mix it with hormone-derived molecules, producing urine that is both metabolic byproduct and social message. The ducts leading from these glands open just below the eye stalks, strategically positioned so that the lobster can aim its plume toward another's sensory organs. It is, in effect, a chemical conversation conducted face-to-face. At a deeper level, these signals intertwine with neurology. Serotonin and dopamine, key neurotransmitters in vertebrates, also regulate aggression and dominance in lobsters. Elevated serotonin levels correlate with boldness and victory. Lower levels accompany retreat. The concentrations of these molecules influence the composition of the urine plume, creating feedback between brain chemistry and social signaling. What a lobster excretes is, quite literally, a reflection of its emotional state, a biological autobiography written in scent. To a human observer, the spectacle borders on surreal. Two armored crustaceans poised in the dark, exchanging invisible clouds that decide their fates. Yet this system exemplifies a profound truth about life underwater. When vision fades and sound travels poorly, chemistry becomes the dominant medium of communication. The sea is, in essence, a chemical world, and the lobster has mastered it better than most. Beyond their battles and courtships, these creatures' chemical exchanges shape entire ecosystems. Territorial marking influences population density. Pheromonal signaling regulates mating cycles. Aggression modulated by scent prevents unnecessary mortality. The result is a finely balanced society governed not by sight or sound, but by the diffusion of molecules through water. In that sense, lobsters have achieved what many complex species struggle with, a method of coexistence through constant communication, even when that communication carries the threat of war. As dusk settles over the reef, the water cools and the currents slow. The victorious lobster emerges from his shelter, antennae sweeping through the dark. Around him drifts a faint chemical halo, his personal signature, a mixture of victory and vigilance. A smaller rival lingers at the edge of scent range, testing the current, reading the molecules for weakness. None appears. The scent says everything. This territory is claimed. This fight is finished. The smaller male retreats into the shadows, the message received without sound, without sight, without violence. In the endless dialogue of the ocean, the lobster's plume stands as one of nature's most refined instruments of communication, part threat, part love letter, part autobiography in water. It is proof that warfare and romance, aggression and intimacy, are not opposites in nature, but reflections of the same evolutionary art, the ability to speak, even when words do not exist. And so, in the quiet dark of the seafloor, long after the bubbles have settled, the scent of one lobster's message drifts through the water, a reminder that even in silence, life beneath the waves never stops talking. The tide pool is still at first glance, a pocket of water captured by retreating waves, framed by barnacle-studded stone and threaded with ribbons of green algae. The sunlight slants through the surface, scattering into gold ripples that dance across the rocks below. Hidden among shells and seaweed, a starfish creeps forward, slow enough to seem motionless. Its arms slide in deliberate rhythm, tube feet extending and retracting in a choreography driven not by muscles, but by water. This animal, ancient and alien, has neither face nor brain, yet it is one of the ocean's most efficient predators. Its methods are silent, its movements patient, and its weapons, chemical and mechanical, operate on a scale that transforms feeding into a slow siege. Beneath its rugged, calcified skin lies an organism built on hydraulics. The starfish's body operates through a water vascular system, an internal network of canals pressurized by seawater drawn in through a small sieve-like opening called the madreporite. The pressure drives hundreds of tube feet lining the undersides of its arms. Each foot ends in a suction cup that adheres to surfaces with a grip that borders on the miraculous. These aren't true suction devices. Rather, they rely on adhesive secretions that bond temporarily to the microscopic textures of rock or shell. By coordinating thousands of these tiny grips, the starfish moves forward, one subtle contraction at a time. It is less a walker than a living hydraulic array, capable of gripping, pulling, and prying with quiet persistence. Its target today is a mussel clinging to the rock. For the mussel, life is simple. Filter water, stay closed, and trust in the strength of its shell. But the starfish's patience undermines even the tightest defenses. One arm settles over the mussel, followed by another and another. The tube feet attach along the shell's edge, pulling slowly, rhythmically, in a contest of endurance that can last hours. The mussel resists, its muscles tightening, its hinge sealed like a fortress gate. Yet no muscle can maintain contraction indefinitely. The starfish exerts only a small, steady force, enough to tire the mollusk without wasting energy. Eventually, a fraction of a millimeter opens between the shells, just enough for the starfish to begin its real attack. From its central mouth, located on the underside of its body, the starfish pushes outward a stomach, an elastic sac that everts through the narrow gap. What follows is not ingestion, but external digestion. The exposed stomach wraps itself around the muscle's flesh and begins secreting a cocktail of digestive enzymes, proteases, lipases, and amylases that liquefy the tissues where they lie. The muscle is digested alive, its soft parts reduced to nutrient-rich slurry before the shell ever opens. When the process is complete, the stomach retracts, pulling the liquefied meal back inside to be absorbed. It is feeding turned inside out, a method so slow and methodical that it borders on surgical. For the starfish, patience is power. Digestion begins before consumption ever happens. But the starfish's chemistry extends beyond feeding. Its entire body is infused with compounds known as asterosaponins, glycosides with soap-like properties that act as powerful deterrents to predators. These molecules disrupt cell membranes, forming microscopic bubbles that destabilize tissues, much as detergent cuts through grease. To fish or crabs attempting to eat a starfish, the first bite delivers an unmistakable lesson. The taste is foul, foamy, and burning on the gills and mouthparts. Many predators learn to spit the starfish out within seconds, often unharmed but thoroughly dissuaded. It is an elegant defense, chemistry as taste, biology as warning label. The body's bitterness is not incidental. It is a language, one that tells the reef, do not try that again. The asterosaponins serve other roles as well. Some function as antimicrobial agents, protecting the starfish's delicate tissues from infection, especially when damaged during regeneration. Others may deter parasitic larvae or prevent bacterial colonization on its skin. The compounds circulate through the coelomic fluid that fills the starfish's internal cavities, making the entire animal, flesh, skin, and secretions, biochemically hostile. Unlike venom, which is meant to attack, these molecules defend passively, ensuring that few creatures ever test their potency twice. Even humans who handle starfish for extended periods sometimes report mild skin irritation, a faint echo of the deterrent effect that guards the animal from predation. Its mechanical system complements its chemical one. The same water-vascular hydraulics that allow movement also empower the starfish to grip prey with remarkable persistence. When scientists measured the pulling strength of individual tube feet, they found that a single arm could exert enough force to pry open a bivalve's shell over hours without fatigue. Each tube foot operates independently yet synchronously, controlled by a diffuse nerve network spread throughout the arms. There is no central brain, only a decentralized coordination system that responds to tactile and chemical cues. This allows the starfish to adapt dynamically. If one arm is damaged or detached, the others continue working, guided by local feedback. The nervous system is less a command center than a chorus of cooperating parts. Regeneration is another of the starfish's quiet miracles. Lose an arm, and it will grow back. In some species, even a single severed arm containing part of the central disc can regenerate an entire new individual. This capacity relies on pluripotent cells, biological blank slates capable of transforming into muscle, nerve, or connective tissue as needed. The process is slow but reliable, a form of biological resilience that ensures survival even after predation attempts or environmental damage. The same immune responses that protect against infection also orchestrate regrowth, balancing destruction and creation in perfect evolutionary rhythm. At a glance, the starfish seems primitive, an invertebrate relic from deep evolutionary time. Yet its lineage has refined specialization to an art form. Its radial symmetry, often seen as a disadvantage in bilateral worlds, allows equal mobility in all directions, a design suited for a life of slow but deliberate conquest. Its eyes, if they can be called that, are simple clusters of light-sensitive cells at the tips of its arms, capable of detecting brightness and movement, but not detail. Still, that is enough. The starfish navigates not by sight but by touch, chemistry and flow. Every inch of its skin is sensitive to dissolved cues in the water, amino acids from prey, reproductive pheromones, and even alarm molecules released by wounded animals. It moves toward opportunity and away from danger, guided by gradients invisible to us, but as clear to it as trails of scent in the air. The ecological role of the starfish is equally profound. On rocky shores and coral reefs, they are keystone predators, shaping communities by regulating populations of mussels, barnacles, and corals. When starfish are abundant, they prevent any single species of prey from dominating, maintaining biodiversity through pressure and balance. Remove them, and ecosystems shift rapidly. Mussels overgrow rocks, corals lose space, and diversity plummets. Their feeding may seem destructive, but it is also creative, opening space for new life to flourish. Even their chemical defenses ripple outward, influencing predator behavior and competition patterns among reef species. The starfish, slow and silent, orchestrates the living structure of the seafloor more profoundly than its pace would ever suggest. Predators that dare challenge it often pay the price. Some triggerfish and large snails specialize in consuming starfish, developing resistance to the toxins or targeting the less protected underside. But even they face a chemical gauntlet. The asterosaponins cause their stomach linings to foam and burn, and many abandon a meal after the first few bites. Evolution's answer is compromise. A few species willing to endure the cost keep the starfish in check, while the rest of the reef population learns avoidance through generations of instinct. It is an arms race fought at molecular level, bile, foam, and deterrent scent replacing claws and speed. In the larger oceanic cycle, the starfish's chemistry extends beyond survival. When it dies, its tissues release those same saponins into the water, where they break down organic matter and deter scavengers. The compounds eventually degrade, feeding microbial communities that recycle nutrients into the ecosystem. Even in death, its molecules influence the chemistry of life around it, a final act of participation in the ocean's metabolic symphony. As evening settles, the tide pool darkens. The starfish remains motionless, its arms draped across the half-empty shell of the mussel. The water, now still and glassy, reflects the fading glow of the sun as plankton drift lazily above. Beneath that calm surface lies a quiet mastery of biology, hydraulics that rival engineering, chemistry that dictates behavior, and patience that conquers movement. The starfish has no eyes to witness its success, no brain to comprehend it, yet its design speaks of an intelligence deeper than thought, an intelligence written in structure, in chemistry, in evolution's timeless calculus. Here, survival is neither speed nor sight, but the ability to turn stillness into strategy, to turn the simplest act of feeding into a display of biochemical warfare. The starfish, both predator and chemist, lives as a testament to an ancient truth of the sea, that the most silent hunters often wield the most sophisticated weapons. The reef is calm until something moves beneath the coral, a flicker, barely perceptible, like the twitch of light on glass. Then, with a motion faster than the blink of an eye, a small blur strikes forward. The impact produces a sharp crack that echoes through the water like the pop of a gunshot. A snail shell nearby fractures cleanly in half, releasing a small cloud of dust and organic debris. The hunter emerges, rainbow armor gleaming in the filtered sunlight, limbs folded like loaded crossbows. This is the mantis shrimp, an animal so powerful that it defies intuition. Its weapon is not just muscle and leverage, but chemistry, physics, and evolution intertwined into one of the most violent acts in the ocean. The mantis shrimp's body is an engineering marvel, compact yet lethal. Its raptorial appendages, modified forelimbs, are sheathed in chitin, reinforced by a mineral composite of hydroxyapatite and amorphous calcium phosphate. This exoskeletal architecture provides a unique balance of hardness and elasticity, allowing it to store and release mechanical energy without shattering. The limbs operate like biological spring-loaded hammers. Flexor muscles in the thorax draw the appendages backward, compressing a saddle-shaped structure of cuticle until it locks. When released, the stored energy propels the club forward at speeds exceeding 20 meters per second, comparable to a bullet leaving a handgun, but underwater, against the drag of seawater itself. At that speed, the strike generates forces surpassing 1,000 times the shrimp's own body weight. But the physical blow is only half the story. As the club accelerates, it creates a region of extreme low pressure immediately ahead of it. The water there boils instantaneously, not from heat, but from pressure collapse. Tiny vapor-filled cavities form in the wake of the strike, a phenomenon known as cavitation. These bubbles collapse in microseconds, releasing bursts of energy that produce heat spikes exceeding several thousand degrees Celsius, momentary temperatures comparable to the surface of the sun. The collapse emits a visible flash of light, a phenomenon known as sonoluminescence. It's an explosion contained within a droplet, invisible to the naked eye but catastrophic to the prey. The prey, often a snail, crab or clam, experiences a double impact. First from the physical strike, then from the cavitation bubble's collapse. The second strike is almost invisible but equally destructive. When the bubble implodes, it releases a shockwave and a spray of reactive radicals, short-lived chemically aggressive molecules like hydroxyl ions that attack organic matter. This means that even if the initial blow doesn't fracture the shell, the collapsing bubble can cook, oxidize, and chemically erode the prey's tissue. It is physics turned biochemical. The mantis shrimp is not merely smashing its meal, it is creating a micro-environment of energy so intense that the water itself participates in the kill. From a biological standpoint, the fact that the mantis shrimp survives its own violence is almost as astonishing as the attack itself. The dactyl club, the striking portion of the limb, is layered in a helicoidal pattern, where microfibers of chitin and mineral twist gradually through the structure like the layers of a laminated shield. This helicoidal architecture deflects cracks and disperses energy across the material, preventing catastrophic failure even after thousands of strikes. Studies have shown that the club's internal fibers are arranged at angles that mimic advanced human composite materials, so much so that engineers have copied this design in developing impact-resistant ceramics and body armor. Evolution perfected this armor long before humans understood the mathematics behind it. At the microscopic scale, each strike leaves scars, microfractures that spread through the upper mineral layer. But these are not flaws. They are part of the shrimp's repair strategy. The damage triggers biological remodeling, with new mineral deposition restoring integrity between molts. Unlike typical crustaceans, the mantis shrimp molts infrequently because regrowing its striking apparatus takes enormous energy. During this time, it hides in burrows or crevices, avoiding confrontation. When the new exoskeleton hardens, it returns to its hunting grounds, armed once again with clubs that can crack shells, shatter glass aquariums, and even injure careless human handlers. The visual world of the mantis shrimp is no less extraordinary than its weaponry. Its eyes, perched on independent stalks, contain up to 16 types of photoreceptor cells, 4 times as many as humans. This allows it to perceive polarized light and wavelengths ranging from ultraviolet to infrared. The shrimp can detect subtle changes in reflection, polarization, and pattern that reveal prey even when concealed by sediment or camouflage. This sensitivity likely aids its targeting system, allowing precise strikes on the weakest structural points of a shell. The combination of visual acuity and ballistic mechanics makes it one of nature's most accurate hunters. It doesn't just see prey, it calculates its destruction. The strike itself begins long before impact. The mantis shrimp positions its body in a coiled stance, adjusting the angle of its clubs relative to the prey. Hydrodynamic studies have revealed that the motion involves a coordinated release of energy from both muscular contraction and exoskeletal spring recoil. When the latch disengages, the club swings forward, accelerating faster than any known muscle-powered movement in the animal kingdom. The water boils in the milliseconds that follow. The prey's shell deforms under compression, and the cavitation bubble blooms like a ghostly sphere before collapsing into a pinpoint of light and force. To an observer filming at tens of thousands of frames per second, the sequence is mesmerizing. A pulse of light followed by a bloom of shattered fragments. This combination of mechanical and chemical destruction is so extreme that scientists describe it as a hybrid weapon system, one that merges kinetic and reactive energy. In practical terms, the mantis shrimp weaponizes physics itself. It is, in effect, an underwater alchemist, turning motion into heat, pressure into plasma, and water into both hammer and fire. Even the audible pop associated with the strike is not the sound of impact, but of bubble collapse, a shockwave that resonates through the reef. Divers sometimes hear it faintly while exploring coral beds. unaware that those small explosions signal the presence of a predator operating at the edge of biological possibility. Beneath this violence lies remarkable ecological balance. The mantis shrimp's predation regulates populations of hard-shelled invertebrates, keeping snails, crabs, and clams in check. Without this pressure, reefs could become overrun with grazers that outcompete corals for space. The shrimp's burrows, excavated in sandy bottoms or within dead coral heads, also create microhabitats for smaller organisms, gobies, worms, and crustaceans that take shelter in the tunnels it leaves behind. Its aggression, though infamous, sustains ecological diversity. Even its waste contributes calcium carbonate and organic matter back to the environment, recycling the very minerals it shatters. Despite its fearsome reputation, the mantis shrimp's aggression is selective and strategic. Territorial battles between individuals are governed by ritualized displays before violence. Two rivals will square off, raising their clubs, flaring their antennae, and flashing their iridescent armor in a test of dominance. Often, the mere display of readiness suffices to settle disputes without a strike. The energy cost of actual combat is immense, and injury can be fatal, especially if the club fractures. When fights do occur, they are brief and explosive, with each shrimp gauging its opponent's capability before retreating. The outcome is decided in milliseconds, yet the balance of power in the reef may shift because of it. From an evolutionary perspective, the mantis shrimp's weapon represents a pinnacle of biomechanical innovation. Its lineage dates back nearly 400 million years, surviving mass extinctions and shifting seas. The earliest forms already bore primitive raptorial limbs, but over time, natural selection honed them into specialized tools for smashing or spearing, depending on the species. Spearers evolved sharp, barbed limbs for impaling soft-bodied prey, while smashers developed the iconic clubs capable of cracking armor. Both designs exemplify divergent evolutionary solutions to the same problem: how to dominate in a world where hard shells and defenses define survival. The chemical side of their strikes, the radical chemistry created by cavitation, remains a frontier of marine physics. Laboratory experiments have confirmed that the collapse of cavitation bubbles can generate transient hydroxyl radicals, which are among the most reactive molecules in nature. These radicals can oxidize proteins, lipids, and nucleic acids in prey tissue, contributing to the lethal effect beyond mere mechanical damage. In essence, the mantis shrimp harnesses a microchemical storm to complement its physical attack. It is as though the creature has evolved to exploit the very laws of thermodynamics for its own predatory advantage. And yet, for all its violence, there is precision. The mantis shrimp rarely wastes energy on fruitless strikes. It studies its prey, probing with antennae, rotating its body to calculate angle and distance, sometimes even tapping the surface to assess structural integrity. Each punch is premeditated, directed at the weakest point. In the seconds before impact, every neuron and muscle fiber coordinate in perfect synchrony. Then comes the strike, so brief that physics, chemistry, and biology merge into a single event of devastating clarity. In the end, the aftermath is quiet. The mantis shrimp retrieves the shattered remains, carefully extracting the soft tissues within. Around it, the reef resumes its rhythm, wrasses darting between corals, plankton drifting in the current, the hum of life continuing as though nothing happened. But in that silence, a truth lingers. Nature's most violent moment is not chaos, it is precision. The mantis shrimp, in its iridescent armor, has mastered the art of controlled destruction, turning the fabric of physics itself into a weapon. In its strike, water becomes fire, bubbles become bullets, and silence becomes thunder. It is evolution's perfect punch, one that cooks, cracks, and cauterizes all at once, leaving behind a world momentarily reordered by the smallest explosion in the sea. The coral reef at dawn hums with quiet menace. Shafts of sunlight thread down through the turquoise surface, illuminating a mosaic of life. Corals unfurling tentacles to feed, clouds of damselfish pulsing in synchronized bursts, and the glint of a parrotfish scraping coral with its beak. But then, from the deeper blue beyond the reef wall, a shadow approaches, broad-headed, thick-lipped, and slow-moving. The Napoleon wrasse, or humphead wrasse, enters the scene like a living relic of coral history. Every motion is deliberate, every bite a negotiation between power and poison. This giant wrasser, one of the largest reef fish on Earth, can grow longer than a diver's arm span. Its forehead bulges like an ancient helmet, and its scales gleam in electric turquoise and jade hues, patterned with delicate labyrinthine lines as if painted by a steady hand. But beneath that beauty lies a biochemistry that borders on the supernatural, an internal defense system that allows it to dine on the reef's most toxic inhabitants without harm. While other fish flee from venomous prey, the Napoleon fish bites boldly into spines, stings, and secretions, turning chemical warfare into breakfast. A crown-of-thorns starfish sprawls across a coral head nearby, its surface bristling with long, venomous spines. Each spine drips with saponins, bitter glycosides that can dissolve cell membranes and kill small fish outright. These starfish are ecological saboteurs, consuming living coral polyps at devastating rates. Few predators dare to confront them. But the Napoleon fish glides closer, lips thick and rubbery, eyes steady. With a slow tilt, it positions itself above the star, flexes its muscular jaws, and clamps down. The spines snap audibly, cracking like brittle glass. The fish's leathery lips act as padding, resisting puncture, while layers of dense connective tissue beneath prevent venom penetration. Each jaw movement grinds with relentless precision, crushing not just the spines but the calcified body beneath. In seconds, a creature feared by the reef becomes a meal in progress. The starfish's venom, normally devastating to tissues, barely slows the wrasser. Inside its gut, acids, enzymes, and specialized detoxifying molecules break down the saponins that would rupture most cells. The key lies in biochemistry. The Napoleon fish's digestive tract produces unique proteins capable of binding saponins and neutralizing their surfactant properties. Instead of allowing the toxins to pierce cell membranes, these proteins form stable complexes that pass harmlessly through digestion. Lipid layers lining the stomach wall are unusually resistant to detergent-like effects, an adaptation believed to have co-evolved with its toxic diet. Meanwhile, a cocktail of bile salts and enzymes helps disassemble the starfish's toxic cocktail before it can enter circulation. The resilience doesn't end with starfish. The Napoleon fish's menu includes sea urchins, boxfish, and even small puffers, each carrying toxins that would incapacitate other predators. In particular, its ability to process tetrodotoxin-containing prey hints at modified sodium channel proteins in nerve and muscle cells, molecular tweaks that reduce toxin-binding affinity. Essentially, the fish's own electrical systems have evolved to ignore certain poisons, a defensive feature similar to that seen in pufferfish themselves. Its teeth, set deep in fleshy lips, are not cutting tools but crushing plates. Behind them lies an even more formidable structure, the pharyngeal jaws, a secondary set of bony plates at the back of the throat. These jaws grind coral skeletons, snail shells, and spiny prey into digestible fragments. The design is a marvel of evolutionary engineering, dense bone overlaid with flexible cartilage, reinforced by powerful muscles that drive slow, crushing pressure rather than speed. It is less a bite and more a hydraulic press. After feeding, the fish pauses, hovering mid-water, flexing its gill covers as it exhales. The dissolved chemicals that would harm others are neutralized, filtered, and dispersed into the current. Around it, smaller fish maintain a respectful distance. The Napoleon fish is not an apex predator by ferocity, but by endurance. Its size, immunity, and calm confidence command quiet dominance. The ecological consequences of its diet ripple across the reef. By preying on crown-of-thorns starfish, the Napoleon fish acts as a biological firewall against coral devastation. During outbreaks, when starfish populations explode due to warming waters or nutrient surges, these wrassers help limit the damage by consuming hundreds of individuals. Their appetite for the toxic and the armored restores equilibrium, buying corals time to regrow. In a sense, they are chemical custodians of reef balance, cleaning up the very poisons that threaten the ecosystem's foundation. Yet, like the starfish they consume, Napoleon fish face their own vulnerability. Slow to mature and long-lived, sometimes surviving for more than half a century, they breed infrequently. Females transition into males later in life through sequential hermaphroditism, a strategy that ensures dominance but slows population recovery. As reefs decline and overfishing intensifies, their numbers have dwindled. Ironically, the very qualities that make them resistant to natural toxins offer no defense against human ones. Cyanide fishing, used illegally to capture live reef fish, poisons them easily, their gills absorbing the same chemistry they evolved to resist in prey. Watching a Napoleon fish patrol its territory is like observing evolution in motion. It moves slowly but deliberately, scanning cracks and ledges where urchins hide. When it locates one, it tilts sideways, wedges the spiny body between coral outcrops, and bites with crushing precision. The sound is distinctive, a wet crunch that reverberates through the reef, followed by the faint hiss of dislodged sand. Each feeding event is both a meal and an act of regulation, curbing populations of animals that would otherwise erode the coral base itself. Its behavior shifts throughout the day. In the morning, it hunts actively, exploiting the light that reveals prey shapes and shadows. By midday, it retreats to deeper slopes or channels where currents cool and visibility drops. There, it cleans its scales against soft corals, rubbing off parasites. In the afternoon, it returns to the shallows, sometimes shadowing schools of smaller wrasses or surgeonfish. Despite its bulk, it is not solitary by aggression. It is simply too self-assured to need a group. In the social hierarchy of the reef, few challenge a creature that can bite through spines and ignore venom. The most fascinating dimension of its biology lies unseen, in its cellular tolerance for chemical stress. Researchers studying its tissues have found elevated antioxidant enzymes and cellular repair proteins, suggesting that its body routinely detoxifies reactive compounds produced during digestion. Its liver, proportionally large for a fish of its size, acts as both chemical refinery and defense line. This internal fortification allows it to exploit ecological niches most fish avoid. To the Napoleon fish, toxins are not threats, they are unclaimed resources, and the reef depends on this strange immunity. Without such predators, toxic species like the crown-of-thorns starfish can surge unchecked, stripping coral reefs of life in months. But where Napoleon fish remain, coral cover is higher, starfish outbreaks rarer, and overall biodiversity richer. It is a predator whose strength lies not in aggression, but in resilience. Toward evening, the reef begins to dim. Parrotfish drift toward their sleeping cocoons, encased in mucus to mask their scent. Small wrasses dart into crevices, their colors fading with the light. But the Napoleon fish still moves, slow and assured, over the dim coral heads. Its scales shimmer faintly in the blue dusk, like metal underwater. A lone crown of thorns drifts on the current, its arms curling in sluggish defense. The wrasser closes in, unfazed by the forest of venomous spines. One measured bite, one decisive crunch, and the predator that devours corals becomes prey to a fish immune to its chemistry. It is a cycle of toxins and tolerance, of attack and adaptation, a molecular war waged not through speed or stealth, but through endurance and immunity. The Napoleon fish is proof that in evolution, power is not always about striking harder. Sometimes it is about refusing to be poisoned at all. As the last light fades across the reef, the giant wrasse disappears into the shadowed coral labyrinth, leaving behind silence and the faint swirl of disturbed sand. The reef breathes easier for its passing. Balance has been restored by a creature whose true weapon is not its jaw, but the chemistry of survival written deep in its blood.